Let's start now. Good afternoon, everyone, and welcome to day 3 of the student webinar lecture series by emerging researchers. We are delighted to have all of you join us once again as we continue this 6-day lecture series featuring student-led lectures along with expert talks from international institutions such as UCCS and UPR. Over the past sessions, we explored cancer biology, therapeutics, neuroscience, and neurodegeneration through diverse scientific perspectives. Today, we move into the two interconnected themes, understanding disease biology and drug discovery fundamentals. These sessions will explore how shared molecular mechanisms contribute to different human diseases and how these biological insights can guide therapeutic development and modern drug discovery. To begin today's session, I would like to invite Dr. Sukant for a short opening talk. Welcome, Dr. Sukant.
Teaching is an important part of growing up as a scientist. To be an impactful researcher, impactful communicator, I wish the youngsters presenting today to a wider audience than just their research colleagues all the best. Go ahead and have a great day. Thank you.
Thank you, Dr. Sukant. We now begin Theme 5, Understanding Disease Biology, Connecting Molecular Mechanism Across Human Diseases. Today's lecture will explore how different diseases are often connected through shared molecular, cellular, and systematic mechanisms, helping us better understand disease progression and therapeutic possibilities. Lecture 1 is by Ashley Verghese, who comes from a background in microbiology. Her lecture is focusing on understanding disease biology, connecting molecular mechanisms across human diseases. Ashley, welcome.
Hello everyone, I am Ashley Vargas, and today I will be discussing a very important and evolving concept in modern biomedical science, that is understanding disease biology by connecting a molecular mechanism across human diseases. Traditionally, we tend to study diseases as completely separate entities. For example, cancer is treated as a genetic disease, diabetes as a metabolic disorder, and Alzheimer's as a neurodegenerative condition. However, when we look at the deeper molecular level, we begin to realize something very interesting. These diseases are not entirely different. In fact, they share a very common underlying biological mechanism. This is where the concept of disease biology becomes very important and powerful. Instead of focusing only on symptoms or organs, disease biology helps us understand what is going wrong at cellular and molecular level. By studying these shared mechanisms, we can identify common targets, I understand why diseases occur together, develop better treatment strategies, as well as move towards precision medicine. So, today I'll walk you through the core molecular mechanisms shared across diseases, how these mechanisms drive different conditions, how they interact with each other, and how this knowledge is transforming medicine. So, let us begin by understanding the big picture. When we look at diseases like cancer, diabetes, and Alzheimer's, they appear very different clinically. Cancer involves uncontrolled cell growth, diabetes involves impaired glucose metabolism, Alzheimer's involves memory loss and neural degeneration. But beneath these visible symptoms lies a shared molecular foundation. At cellular level, all diseases arise from disruption in normal biological process, maybe cell signaling, gene expression, protein function, metabolism, or a immune response. For example, inflammation plays a very important role in both diabetes, as well as in cancer. Oxidative stress contributes to both neurodegeneration and cardiovascular diseases. Metabolic imbalance is seen in obesity, diabetes, and even cancer. So instead of thinking in isolated category, we must think in terms of interconnected biological system. This shift in thinking has major implication. It allows us to use the same drug for multiple diseases. This foundation of systems biology, which views the body as an integrated network rather than separate parts. Six core molecular mechanism. So now let us move on to the central part of the seminar. The six shared molecular mechanisms that form the basis of many diseases. These mechanisms are chronic inflammation, oxidative stress, genetic mutation, protein misfolding, immune dysregulation, and metabolic disbalance. So these are not isolated events, they interact with each other and create a complex network that drives disease progression. In the next session, I will explain each of these mechanisms in detail, including how they contribute to different diseases. Chronic inflammation. So let us begin with chronic inflammation. And inflammation is a very natural process and it is a protective response of the body. In acute condition, it helps eliminate pathogen and promotes healing. However, when inflammation becomes persistent, it becomes harmful. Chronic inflammation is characterized by continuous activation of the immune system, even in the absence of infection. This leads to tissue damage over the time. Key molecules involved in inflammation are IL-6, that is interleukin 6, or tumor necrosis factor alpha, or TNF alpha. These cytokines create a self-sustaining inflammatory loop. Now let's just see how these applies to a disease. So, for example, in cancer, inflammation promotes tumor growth by stimulating cell proliferation or suppressing the immune responses. So, looking into the type 2 diabetes, inflammation interferes with insulin signaling, leading to insulin resistance. And in cardiovascular diseases, inflammation contributes to plaque formation in arteries, leading to atherosclerosis. So, inflammation acts as a common driver across multiple diseases. This is why anti inflammatory drugs are being explored as treatment for wide range of conditions. Oxidative stress and genetic mutation. So, next we move on to the oxidative stress. So, it occurs when there is an imbalance in ROS or reactive oxygen species and body's antioxidant defenses. So ROS are highly reactive molecule that can damage DNA, protein, or lipids. This damage accumulates over time and contributes to aging and disease. Looking into the example of some diseases, for example, in Alzheimer's, oxidative stress damages the neuron and in cancer, it leads to DNA mutations. And in Parkinson's, it contributes to neuronal death. This brings us to genetic mutation. So, when does a mutation occur? So, it occurs when DNA is altered. And these changes can lead to activation of oncogenes, disable tumor suppressor gene, and it alters the protein function. So in cancer, multiple mutation accumulates leading to uncontrollable cell growth. When looking into another disease, completely different disease, that is cystic fibrosis, a single mutation in CFTR gene causes systemic disorder. So oxidative stress and mutation are closely linked. Oxidative damage often lead to genetic changes. Together, they form a major driving force in disease development. So, moving on to the next part, that is the protein misfolding and immunodysregulation. So, it is very important. That is because proteins are one of the most common compounds in our body. So, it has to be folded into a specific shape to function properly. When this process fails, misfolded protein accumulates and it forms aggregates. These aggregates disrupt the cellular function. So, when taking an example of a disease, for example, in Alzheimer's, Beta amyloid proteins form plaques, whereas in another neurodegenerative disorder such as Parkinson's, alpha synuclein forms Lewy bodies. These aggregates are toxic to cells, both beta amyloids as well as alpha synuclein, especially neurons. So these are the neurodegenerative diseases. Next, we are going to look into the immune system dysregulation. So we know that immune system must maintain a balance between activation and tolerance. If it becomes overly active, it attacks the body, leading to autoimmune disorders. If it becomes underactive, it fails to eliminate harmful cells, such as cancer cells. So, tumor exploits the immune checkpoints to escape the detection, especially PD1 and PDL1. This understanding has led to many therapies, especially immunotherapy, which reactivates the immune system to fight cancer. So both protein misfolding and immune imbalance plays a critical role in disease progression. Metabolism is not just the energy production, it is deeply connected to disease. So this brings us to the next category, that is the metabolic dysregulation. So metabolic dysregulation occurs when the normal metabolism pathways are altered or disrupted. So some of the pathways that is disrupted in diseases are in diabetes, mainly the insulin metabolism is affected, that is the insulin signaling is impaired, and in obesity adipose tissues promote the inflammation, and in cancer cells undergo metabolic reprogramming. So one classic example is the Warburg effect, where cancer cells rely on glycolysis even in the presence of oxygen. This allows rapid energy production for growth. And metabolism also interacts with other systems. It influences immune response, it affects the inflammation, it contributes the oxidative stress. So metabolism is not isolated. It is the central of disease biology. Now we can look into some of the case studies. When we are bringing everything together, these mechanisms do not act independently. They form an interconnected network. So let us take cancer as an example. Cancer involves genetic mutation, initiating transformation, inflammation promoting growth, immune invasion enabling survival, and metabolic reprogramming supporting proliferation. So these all contribute to cancer. Similarly, when looking into a completely different neurodegenerative disease, let's just take Alzheimer's. So it involves protein aggregation, oxidative stress, and neuroinflammation. These mechanisms reinforce each other, creating a cycle of disease progression. This concept is called disease network convergence. Understanding these shared mechanisms has revolutionized medicine. First, it allows us to target the shared pathway using drugs. Second, it enables the drug repurposing where existing drugs are used for new diseases. Third, it supports a precision medicine where treatment is tailored to individual patient based on molecular profiles. This is the future of healthcare, moving from symptoms based treatment to mechanism based therapy. To conclude the entire presentation, the diseases that appear different on the surface often share common molecular roots. Mechanisms such as inflammation, oxidative stress, mutation, protein misfolding, immune imbalance, and metabolic dysfunction are interconnected. Understanding these connections allow us to develop better treatment, predict disease outcomes, personalize therapy. The future lies in integrated system biology where we study the entire network rather than the isolated pathway. We have come to the conclusion of the entire presentation. Once again, thank you for your attention.
Thank you, Ashley, for that well explained introduction to the theme. Our next Our next lecture is by Sri Deepti. Her lecture is focused on immune system as a central driver of disease biology. Welcome, Sri Deepti.
Hello everyone. Today I will be discussing about immune mechanisms as a unifying framework in disease biology. Most disease discussions are usually separated into categories such as infectious diseases, autoimmune disorders, inflammatory diseases, or immunodeficiency disorders. However, in this presentation, I would like to approach disease biology from a different perspective. Instead of studying diseases individually, I will focus on the immune system as a shared molecular framework that connects multiple diseases through common mechanisms. The immune system is traditionally viewed as a defense system that protects the body from pathogens. But in reality, it is much more than that. It is a highly dynamic and tightly regulated biological network that continuously balances protection and tissue integrity. At its core, the immune system performs three major functions: recognition of molecular patterns, activation of signaling pathways, and regulation of immune intensity. These same mechanisms are involved across multiple diseases. For example, in infections, these pathways help eliminate pathogens. In autoimmune diseases, the same pathways become misdirected towards self tissues. In chronic inflammatory diseases, immune activation becomes persistent. And in immunodeficiency disorders, immune responses become insufficient. So, although the diseases appear clinically different, the molecular mechanisms are often shared. This is why immunology provides such a powerful integrated framework for understanding disease biology. So, in this image, we can see the immune system represented as a central interconnected networks linking infections, inflammations, cancer, and tissue damage. Pathways do not operate independently within one disease category. Instead, the same signaling networks influence multiple pathological conditions simultaneously. One of the most important concepts in immunology is immune homeostasis. The immune system must maintain a balance between effective pathogen elimination and protection of host tissues. If this balance is maintained properly, pathogens are removed efficiently with minimal tissue damage. However, if the immune response becomes excessive, hyperactivation occurs. This leads to excessive cytokine release, persistent inflammation, tissue injury, and chronic disease progression. On the other hand, if immune activity becomes too weak, pathogens are not cleared effectively. This results in recurrent infections, opportunistic infections, and increased disease susceptibility. So, disease outcomes are strongly influenced not simply by whether the immune system is active, but by how well its activity is regulated. This concept connects many diseases under a common mechanistic framework. For example, autoimmune diseases represent excessive or misdirected immune activity. Immunodeficiency disorders represent insufficient immune activity. And chronic inflammatory diseases often involve prolonged activation without proper resolution. Therefore, balance is the central determinant of immune mediated diseases. At the molecular level, adaptive immunity begins with antigen antibody interaction. So, antigen antibody interaction is a specific chemical interaction between an antigen and an antibody during an immune response. Each antibody recognizes a specific region on the antigen known as an epitope through its antigen binding region called the paratope. This specificity is extremely important because it determines what the immune system targets and what it ignores. The interaction depends on multiple non-covalent forces, including hydrogen bonds, ionic interactions, hydrophobic interactions, and van der Waals forces. Individually, these forces are weak. However, collectively they produce highly specific and stable binding. This is a very important biological principle because immune specificity depends on structural complementarity. Even very small molecular changes can significantly alter immune recognition. Antibody binding is not simply a recognition event, it initiates multiple downstream immune responses. As you can see, antigen antibody interaction progresses through several stages. First, formation of antigen antibody complexes. Second, visible reactions such as agglutination and precipitation. And third, neutralization or destruction of antigens. These interactions produce several important biological effects. First, neutralization blocks pathogens or toxins from interacting with host cells. Second, agglutination and precipitation help cluster antigens together, making them easier to eliminate. Third, opsonization enhances phagocytosis by marking pathogens for immune cell recognition. Finally, antibody binding activates the complement system, which amplifies immune responses further. So, a single antigen recognition event can initiate a complex cascade involving multiple immune mechanisms simultaneously. The complement system is one of the most important amplification mechanisms in immunity. It consists of serum proteins that interact through catalytic cascades to eliminate pathogens and regulate immune responses. There are three major pathways of activation. The classical pathway, which is triggered by antigen antibody complexes, the lectin pathway, which is triggered by microbial carbohydrates, and the alternative pathway, which is activated directly on microbial surfaces. Although initiation differs, all three pathways converge at the formation of C3 convertase and later C5 convertase. These enzymatic complexes amplify the immune response dramatically. The major effector molecules include C3b, which promotes opsonization, C3a and C5a, which function as inflammatory mediators, the membrane attack complex or MAC, which forms pores in microbial membranes and causes lysis. This demonstrates how the immune system converts localized recognition into systemic defense. An important concept in disease biology is that protective mechanisms can become pathogenic when excessively activated. Although complement activation is essential for host defense, uncontrolled complement activity can damage host tissues. For example, excessive C3a and C5a production causes strong inflammatory responses. Next, persistent neutrophil recruitment increases tissue injury, and endothelial damage contributes to chronic inflammation. So, the same mechanisms that eliminate pathogens can also drive disease pathology if regulation fails. This concept is highly relevant across many inflammatory disorders. It again reinforces the theme that disease biology is often determined by dysregulation of shared molecular pathways. Immunodeficiency disorders arise when immune mechanisms fail to function effectively. These disorders are classified into primary and secondary immunodeficiencies. Primary immunodeficiencies are caused by inherited genetic defects. Secondary immunodeficiencies occur due to external factors such as infections, malnutrition, chemotherapy, or immunosuppressive therapies. The major consequence is increased susceptibility to infections. Importantly, organisms that are normally harmless can become life-threatening in immunocompromised individuals. This demonstrates another major principle in disease biology. The severity of disease is not determined only by the pathogen itself, but also by the functional state of the immune system. Now, I will discuss about severe combined immunodeficiency or SCID, which is one of the most severe forms of primary immunodeficiency. SCID occurs due to genetic defects that impair lymphocyte development, leading to absence or dysfunction of both T cells and B cells. This means both humoral immunity and cellular immunity are severely affected. This slide highlights four important molecular mechanisms involved in SCID. The first is defective cytokine signaling. In this condition, developing immune cells cannot properly receive growth and survival signals, so they fail to mature. The second mechanism is ADA deficiency, where toxic metabolic waste accumulates inside developing lymphocytes and destroys them before maturation. The third mechanism involves defects in DNA recombination, especially mutations in RAG genes. Normally, T cells and B cells rearrange their DNA to form antigen receptors. If this process fails, functional immune receptors cannot be formed. The fourth mechanism is intracellular signaling defects, where immune cells may be present, but they cannot activate properly because signaling pathways are defective. Even though these mechanisms are different, the outcome is similar. Severe impairment of adaptive immunity, recurrent infections, and failure to mount effective immune responses. So as you can see in this image, this is the famous kid bubble boy. This image represents children with SCID who had to live in highly sterile isolation environments because even common microorganisms could become life-threatening. Since these patients lack proper immune protection, normal environmental exposure becomes extremely dangerous. The bubble boy became one of the most recognized clinical examples showing how a molecular immune defect can affect the entire body and completely alter survival. This slide focuses on HIV as an example of acquired immunodeficiency. Unlike SCID, HIV is not inherited. Instead, it develops due to viral infection. As shown in the slide, HIV specifically targets CD4 positive T cells, which are very important for coordinating immune responses. The progressive depletion of these cells leads to impaired immune regulation. This slide highlights three major consequences of CD4 T cell loss. First, immune coordination becomes impaired because helper T cells normally activate multiple other immune cells. Second, activation of cytotoxic T cells and B cells decreases, reducing both cellular and humoral immune responses. Third, susceptibility to opportunistic infections increases significantly. As the disease progresses, severe immune collapse can eventually lead to AIDS. An important point mentioned in the slide is that pathogens can hijack immune mechanisms to promote disease progression. So, HIV demonstrates how disruption of one major immune cell population can affect the entire immune network. Now, the slide brings together all the concepts discussed throughout the presentation. As shown in the slide, the same core immune mechanisms are repeatedly observed across different diseases. These include molecular recognition through antigen-antibody interactions, signal amplification through the complement system, functional responses such as phagocytosis, lysis, and inflammation, and regulation of immune intensity and duration. The disease outcomes depend on the strength of immune activation, duration of response, accuracy of targeting, and efficiency of regulation. So we come to an understanding that when immune responses are balanced, they provide protection. But when they become excessive or insufficient, they can lead to inflammation, tissue damage, or immunodeficiency. So, the main takeaway is that different diseases are not driven by completely different systems, but are often different outcomes of the same immune system. Thank you.
Thank you, Deepti, for that well explained overview of the immune system's role across human diseases. And building upon these concepts, we now move towards cellular stress biology. Our next lecture will be delivered by Shreya Das, who comes from a academic background including biological sciences and tissue engineering for regenerative medicine. Her lecture is focused on cellular stress responses, the common failure mode across human diseases. Over to you, Shreya.
Then everyone. Thank then everyone. Thank you for being here. My name is Shreya Das, and today I will be taking you through my lecture titled Cellular Stress Responses, the Common Failure Mode Across Human Diseases. So I'm going to give you a systems level view of adaptation, fragility, and therapeutic collapse. This is going to be a systems level lecture. Here, we are not going to catalog pathways. Rather, we will build a single unified argument that the same adaptive machinery cells use to survive stress is also the machinery that drives disease when it is chronically overloaded, co-opted, or even pushed past the point of recovery. By the end of this session, I want you to be able to look at cancer or at any antibiotic resistant infection or at a neurodegenerative disease and immediately recognize the common operating system underneath all three. So let us begin. Before we go into any specific pathways, I want to establish the conceptual lens for this entire lecture. So here, look at the four statements on this slide. Stress pathways are conserved survival circuits. The same systems that protect can fail under sustained load. Disease progression tracks how close a cell is to its adaptive threshold. And cancer, infection, and neurodegeneration all exploit the same stress logic. The last point is the one I want you to anchor on because this is the claim that makes the entire lecture worth giving. We tend to teach diseases in separate silos. Oncology here, microbiology there, neuroscience somewhere else. But when you zoom in to the molecular level, you find that all these three disease categories exploit and even exhaust the same conserved survival programs. The unfolded protein response, redox buffering, programmed cell death pathways. The same circuits go on again and again. So the question here I'm asking you is to start asking from this slide onward is not which pathway is activated. Rather, the question should be how close is this system to its tipping point? How much adaptive reserve remains? And who is closest to that edge? That framing will change how you think about both disease and therapy. So let us now build that argument from the ground. First, Let's start with the endoplasmic reticulum. As we all know, the ER is the cell's primary protein quality control compartment. Every secreted and membrane protein has to pass through it. Under normal conditions, the ER maintains a careful balance between the rate of protein synthesis and its folding capacity. But when the balance tips, that is, when the misfolded proteins accumulate faster than they can be processed and cleared, the cell activates the unfolded protein response, that is, the UPR. Now, I want you to think of the UPR not as a pathway, but as a decision program. It senses the severity and duration of the stress load and it makes a choice. Do we adapt and survive, or do we trigger apoptosis and eliminate the compromised cell altogether? In tumors, especially those living inside the hypoxic and nutrient-poor microenvironments, the UPR is hijacked. Malignant cells learn to keep the adaptive arm of the UPR continuously active, which helps them maintain the proteostasis and survive conditions that would otherwise kill a normal cell. In the same manner, in chronic neurodegenerative diseases, on the other hand, the same machinery gets overloaded for years. The UPR cannot restore balance because the misfolded protein burden never gets resolved. And at that point, the very response that was meant to protect the cell now starts to contribute to its death. So here we see that the same system gives opposite outcomes. What determines it? The context. Three branches govern this basic decision. It is PERK, IRE1 alpha, and ATF6. So we are looking at each of them now. The three kinases and the downstream effectors are not redundant. Rather, they are layered. Each one responds to ER stress in a very distinct manner. And together, they constitute a modular adaptive architecture. So let us take each one briefly. PERK. PERK is activated first, and its most immediate action is to phosphorylate EIF2 alpha. This halts the most cap-dependent translation. It slows the entire cell down, reduces the incoming load on the ER. But simultaneously, PERK also allows the selective translation of ATF4, which drives transcription of genes involved in amino acid metabolism, antioxidant defense, and cellular survival. In tumors, this arm is particularly significant for surviving glucose limitation and hypoxia. Now coming to IRE1 alpha. It has got a very unique enzymatic activity. It acts as an endoribonuclease that splices the XBP1 mRNA. The spliced form of XBP1, called as the XBP1S, is actually a transcription factor that massively expands the ER's folding and secretory capacity. It upregulates chaperones, ER-associated degradation components, and lipid biosynthesis pathways. This arm is especially important in cells with high secretory demand, as in plasma cells, for example, or even in tumor cells producing large amount of pro-survival factors. ATF6 takes a different route. Under ER stress, it translocates to the Golgi apparatus, where it is cleaved by site 1 and site 2 proteases to release an active transcription factor. This drives the expression of VIP, GRP94, and other major chaperones that help restore the ER homeostasis. Now, here comes the most important translation point. All the three arms are context dependent, as we have already seen. The same signaling that actually protects a tumor cell under acute hypoxia can, if sustained long enough, flip into a pro-apoptotic program. CHOP is a transcription factor downstream of PER. It can give the best studied example of this kind of a switch. However, we'll see this flip in action when we move to neurodegeneration. First, coming to cancer. How specifically does a tumor exploit this UPR machinery? As we all know, solid tumors create a profoundly hostile internal environment. As the tumor grows beyond the reach of its existing blood supply, regions become hypoxic. Glucose and amino acids are depleted. Reactive oxygen species accumulate from metabolic activity. The pH drops as lactic acid builds up. So each of these conditions, as we discussed, hypoxia, nutrient deprivation, ROS, acidosis, each is an independent trigger of this ER stress. Tumor cells therefore face a continuously elevated ER stress burden. As a result, the UPR has to be essentially always on. And this is what the cancer cells adapt. They learn to maintain the adaptive arms of the UPR chronically active while suppressing or even delaying the pro-apoptotic switch. The result is a cell that can tolerate conditions that would kill any normal epithelial cell. Practically, this means UPR activity supports malignant cell proteostasis. It preserves translation control, so the cell can continue making the proteins it needs for survival. It activates autophagy, which recycles the damaged organelles and proteins to extend viability under nutrient stress. And it even upregulates anti-apoptotic proteins like BCL2. The key conceptual point that I'm trying to establish here is that tumors do not invent new biology. Rather, they redirect and sustain a conserved repair program that already exists in every cell. They just keep it running past the point where normal cells would switch it off. And that co-option, as we'll see in the second half of this lecture, is also a vulnerability. Now coming to neurodegeneration. I want you to hold everything I just said about cancer and now flip it completely. In neurodegeneration, cells don't successfully exploit the UPR. They fail to sustain its adaptive output long enough to survive. The core problem is chronic, unresolvable protein misfolding. In diseases like Alzheimer's, Parkinson's, ALS, and even prion disease, specific proteins form aggregates that resist the cell's clearest machinery. They accumulate in and around the ER. They maintain the ER stress continuously for months or years. At first, the UPR tries to compensate. Chaperones are induced. Translation is slow. ERAD, that is, the ER-associated degradation, is upregulated. These are exactly the same protective responses we have just described in the tumor cells. But we should always keep it in mind that neurons are post-mitotic. As a result, they cannot dilute the aggregate burden by dividing. They cannot replace themselves. And over time, this continuous ER stress buildup, the load, exceeds what any adaptive response can manage. So when that happens, the UPR does not simply stop working. It shifts. The PERK arm, which was once protected translation, now increasingly produces CHOP. The caspase cascades get activated. The same machinery that provided a survival window in case of acute stress now contributes to the lethal outcome. So this is the critical conceptual point for this slide and in fact for the whole lecture. The same UPR circuit that a cancer cell uses to thrive is the circuit that the neuronal cells run until it exhausts itself and they have to die. The biology is conserved, the outcome depends entirely on the duration and context of the stress. Now we have to move from proteotoxic stress to a second conserved stress system, that is the oxidative stress. As we all know, reactive oxygen species or the ROS, they are not inherently pathological. They are a normal feature of cellular metabolism generated primarily by electron transport and NADPH oxidases. At low to moderate levels, they function as signaling molecules. They activate growth factor pathways, regulate immune responses, and modulate transcription factor activity. But the problem begins when the ROS production exceeds antioxidant buffering capacity. At that point, they become the damage drivers, oxidizing proteins, peroxidizing membrane lipids, and even damaging DNA. What makes ROS so interesting from a systems biology perspective is that the same chemistry is used differently across disease contexts. In innate immunity, macrophages deliberately generate massive oxidative bursts far beyond any incidental metabolic level as a frontline antimicrobial weapon. This is intentional ROS production. But the macrophages must simultaneously protect themselves from their own chemistry using the internal redox buffers. Pathogens in turn have evolved structural antioxidant defenses to survive that oxidative attack. And tumors generate high ROS internally through oncogenic metabolism, then buffer it using transcriptional antioxidant programs. So as we see, it is the same chemistry. Okay, three completely different operational contexts, and all three are exploitable therapeutically. Let me now make the infection context specific because it is one of the most instructive illustrations of stress biology as a therapeutic opportunity. When a macrophage engulfs a pathogen, it activates the NADPH oxidase on its phagosomal membrane. This produces superoxide, which rapidly gets converted to hydrogen peroxide and then to the highly bactericidal hypochlorous acid. The resulting oxidative burst can kill most bacteria within minutes. But our case study here is MRSA, that is methicillin-resistant Staphylococcus aureus. It has evolved a remarkably effective structural defense against this. It produces a golden carotenoid pigment called staphylozanthin, which is a potent antioxidant. Staphylozanthin quenches the ROS before they can damage the bacterial membranes or proteins, giving MRSA the significant resistance to phagocyte-mediated killing. The key word here I want you to focus on is structure. So this is not a transcriptional response. It is a constitutive antioxidant core built already into the bacterial cell wall, and its biosynthesis depends on a specific enzyme called CRTM, that is dehydrosqualene synthase. But what makes this therapeutically exciting is the following. Like suppose if you inhibit the CRTM, that is if you disarm the staphylozanthin shield, you do not directly kill the bacteria. You simply remove its antioxidant armor. The bacterium then becomes vulnerable to the host immune response it would otherwise survive. And because you're not generating direct bactericidal pressure, you are also not selecting for the classical antibiotic resistance in the same way. That is actually the logic of antivirulence therapy, and it is directly rooted in stress response biology. Now, let us move to how tumors handle their own internal ROS burden. Cancer cells are in a metabolic paradox. Oncogenic mutations in pathways like RAS and MYC, they drive elevated metabolic activity, increased electron transport, and chronically elevated mitochondrial ROS production. That ROS would normally be cytotoxic. But we should look into how do tumors survive their own chemistry. The answer very frequently is the NRF2. So what is NRF2? NRF2 is the master transcription factor of the entire antioxidant response. Under normal conditions, it is held in the cytoplasm and it is continuously targeted for proteasomal degradation by its repressor protein KEAP1. KEAP1 senses oxidative stress through reactive cysteine residues, and when the ROS levels rise, KEAP1 is modified. This releases the NRF2, which then translocates to the nucleus, and it activates a broad program of antioxidant gene expression. The NRF2 target genes include HO1, NQO1, GCLC, which synthesizes glutathione, and TXNRD1. Together, these form a powerful antioxidant network that reduces ROS back to tolerable levels. In many cancers, ROS keep is mutated or lost and NRF2 is constitutively active. The result is a tumor cell with a permanently elevated antioxidant program, one that buffers both its endogenous ROS production and the oxidative damage from chemotherapy and radiotherapy. That is why NRF2 activation is so commonly associated with treatment resistance. It is not just protecting the tumor from its own metabolism, rather it is protecting itself from us. We are now entering the translational section of this lecture. Everything I have described so far has been about how the disease cells manage stress. Now I want to turn that around and ask you people, how do we exploit this? The central insight is this. The disease cells, that is the tumor cells, therapy-resistant bacteria, chronically stressed neurons, they are not sitting comfortably in a homeostatic equilibrium. They're already operating near the edge of their adaptive capacity. The stress buffering circuits are already heavily engaged. That means a relatively small perturbation, one that a normal cell with full adaptive reserve can tolerate easily, that is enough to push a disease cell past its tipping point into irreversible failure. This is the logic of threshold directed therapy. So you're not simply blocking a growth pathway. Rather, you're identifying where the adaptive margin is narrowest and delivering a stress load the cell cannot absorb. The two most important cell death programs in this context are ferroptosis and apoptosis. Ferroptosis is triggered when antioxidant capacity fails, specifically when GPX4 is lost or even glutathione is depleted, allowing iron-catalyzed lipid peroxidation to escalate uncontrollably. Apoptosis can be triggered by pushing unresolved ER or proteotoxic stress past the CHOP threshold we discovered earlier. The therapeutic goal is selective. Cross the threshold in disease cells which have less reserve before you cross it in the normal cells which already have more. Now let's make ferroptosis mechanistically precise. So what is ferroptosis actually? Ferroptosis is a regulated form of cell death driven by iron-dependent lipid peroxidation. It is distinct from apoptosis, necrosis, and even autophagy. It does not involve caspase activation. It does not depend on the mitochondrial outer membrane permeabilization. Its execution is anchored to the plasma membrane and it proceeds through the uncontrolled accumulation of oxidized phospholipids. The central gatekeeper here is the GPX4, that is the glutathione peroxidase 4. GPX4 is unique among the glutathione peroxidase family because it directly reduces the phospholipid hydroperoxides. That is, the oxidized lipids embedded in the membrane binder. So while GPX4 is active and the glutathione supply is adequate, the lipid peroxides are neutralized as fast as they form and the membrane remains intact. But the situation gets changed when the GPX1 is inhibited by compounds like RSL3, for example, or when glutathione is depleted by blocking the cysteine import through the system Xc minus transport. That is, then the peroxide neutralization fails. Iron, acting through the Fenton chemistry, catalyzes the propagation of lipid peroxidation across the membrane. This results in the progressive destruction of the bilayer, and as a result, the cell ruptures. The reason ferroptosis matters therapeutically is the context. The tumor cells that already carry high oxidative burden, high lipid peroxidation pressure, or mesenchymal or therapy tolerant phenotypes are operating within the GPX4 glutathione system near full capacity. In those cells, a small additional push, GPX4 inhibition, or even glutathione depletion, that is sufficient to cross that therapeutic threshold. In normal cells with more antioxidant reserve, the same dose does not cross it. So that differential is actually the therapeutic window we are trying to open. Till now, we have already established what to target. But before I close, I want to briefly address how to target. Identifying a stress response dependency is necessary, but that is not sufficient for drug discovery. We also need to know whether the target has got a binding site, a pocket, a catalytic residue, allosteric surface that a small molecule can actually engage. This is where the structural biology and the computational methods enter the workflow. AlphaFold 2 has fundamentally changed early stage target assessment. Before AlphaFold, we often had to wait years for an experimental crystal or a cryo-EM structure before we could even ask whether a target has got a druggable pocket or not. AlphaFold now provides high confidence structural predictions for virtually any protein in the human proteome and for many pathogen proteins as well. That accelerates the triage enormously. Once a structure is in hand, molecular docking allows us to computationally explore which compounds might fit into the identified pockets and how. This is obviously not a substitute for experimental medicinal chemistry, but it is a powerful early prioritization tool that can reduce the number of compounds that need to be synthesized and tested. But I want to flag one critical limitation that is often underestimated. That is the conformational state selection. Because proteins are not static, a predicted structure captures only one conformation. But the biological relevant conformation, that is the one that the protein adopts under disease conditions or upon ligand binding, can be different as well. So choosing the wrong conformation to dock against or can lead to false positives and even missed opportunities. So we can say like structure is the starting point, but it is not the finished blueprint. Let me now bring everything together. The central argument of this lecture is on the slide. The UPR, redox buffering, and programmed cell death pathways are not separate features of the separate diseases. Rather, they are a single, conserved, adaptive operating system deployed again and again in slightly different configurations across cancer, infection, and neurodegeneration. Where these diseases differ is not in the biology, it is in the position along the stress response curve. Cancer finds a way to sustain itself near the adaptive age, co-opting the stress programs to survive conditions that should be lethal. Infection uses structural antioxidant architectures to persist under immune pressure. Neurodegeneration reflects what happens when the same adaptive system is pushed for too long without resolution. It runs out of capacity and as a result, the cell collapses. And therapeutically, all three of those positions can be exploited. In each case, the strategy remains the same. Identify where the adaptive reserve is narrowest and deliver a targeted stress load that crosses the threshold in the disease cell before it crosses it in the normal tissue. That is the translational promise of stress biology. And that is what my third lecture will build on. Thank you for your attention. I will leave you with this. That is, across human disease, stress responses are not just compensatory pathways. Rather, they are the common operating system of failure, and the next generation of therapeutics will be defined by how precisely we learn to crash that system in the right cell at the right time.
Thank you, Shreya, for that conceptually rich presentation. We now continue our exploration of disease biology through the lens of epigenetics. Our next lecture will be delivered by Sanampreet Kaur, who comes from a background in human genetics. Her lecture is focused on epigenetics across human diseases. Welcome, Sanampreet.
Hello, my name is Sonampreet Kaur, and I'll be presenting today on the topic epigenetics across human diseases. I want to begin with one of the core paradoxes in the biology, which goes like if every cell in your body is having same genome, how come every cell is so different? The cells are different not only in their structure, they're different in their function, in the protein production, in metabolism, and also in lifespan. There are certain cells like neurons which are not only structurally very different, but they also have exceptionally long lifespan because they are having limited capacity for regeneration. And then there are cells like liver cells which are having quite an amazing capacity for regeneration. And then there are some special cells like muscle cells which have the capacity for change in the shape because of the presence of the contractile proteins like actin and myosin. So these individual differences are not because of the differences in the DNA sequences, because each cell has inherited the same amount of genetic material that was there in the zygote before the embryo got developed. So the paradox becomes even more striking when you compare individuals. So humans share more than 99% of the DNA with each other, and some papers claim this number to be 99.6%, while some claim it to be 99.4%. Whatever be the case, we are more alike in our genome than we had initially considered ourselves to be. Yet we are entirely different when we consider our appearances, or our behavior, our stress responses, or even our susceptibility to diseases. And the similar kind of paradox is also seen during development. So when an egg is fertilized or when zygote is formed, that zygote gives rise to all these cells in your body, right? So until the embryo is at the level of 8-celled stage, each of these eight cells have the capacity to give rise to each and every individual cells and individual tissues in your body. But that doesn't happen. So what we observe is the later during the stage of development, each cells acquire their own fate. Some cells become neurons, some become the bone tissue, some become the vascular tissue, and some become livers or sensory organs, and hundreds of other kind of specialized cell types. This complexity emerges without changing the underlying DNA sequences. And this tells us that the DNA alone cannot fully explain the biological outcomes. Cells require certain systems which will determine how that DNA sequences will function, how they will become activated, they will become silent, or they will be repressed. And this kind of systems are collectively termed what we call the epigenetics. Strongest evidence for the epigenetic mechanisms comes from the twin studies. So we study the identical twins, which are also called as the monozygotic twins because they are basically developing from the same zygote. So they are also inheriting exactly the same DNA. So because they are inheriting the same DNA, it is expected that their biological outcome will also be same, and it will remain same throughout their entire lifetime. But that's not what we observe. So as twins age, the differences gradually start to emerge in their physical health, in their behavior, in their disease susceptibility, their cognitive traits, their psychological traits. And this was shown in one of the important studies conducted by the Asler and his colleagues. So basically, they showed that the young identical twins, as you can see, the 3-year-old twins here, they were showing the similar patterns of the epigenetic tags, which were in the same place. But as they become older, like you can see in the 50-year-old twins, these epigenetic marks are in entirely different places. So the epigenetic marks are not like static entities, they are dynamic, they are constantly changing throughout the life as people are exposed to different environmental conditions. And this is the reason why one twin may develop cancer while the other doesn't, or why one twin may show some kind of metabolic disorder and the other doesn't, or why one twin may show greater resilience to environmental stressors and the other twin is more vulnerable to these kind of environmental stressors. These differences are not associated with the DNA sequences because the DNA sequences in the twins is essentially similar. They are differing in the exposure to different environmental factors like diet, stress, toxins, or physical activity, social experiences, and these environmental factors in turn alter how these genes will be expressed or they will alter the gene expression. And because of alteration in the gene expression, the biological identity or the phenotypic outcome will be entirely different. And this outcome may not be apparently different during the early years of life, but gradually as the twins become older, the differences become to diverge even further. We've seen the paradox and the evidence from the twin studies. We can formally define what epigenetics is. Now, epigenetics is often defined as the heritable and reversible changes in gene expression that occur without changing the underlying DNA sequences, which means that the DNA code will remain the same while the activity of the genes will change. And there are three major ways in which this can happen. There can be gene activation, which basically means that the gene will be more accessible for the transcription. The part of the DNA is transcribed to mRNA by a process called transcription. When this process can happen smoothly, when the part of the DNA is accessible for certain proteins which are uh, allowing transcription to take place, the gene will be expressed or the gene will be activated. So messenger RNA will be produced, which will later be read, and a protein will be synthesized, which will be folded into a 3D conformation. So this process will readily happen. On the contrary, gene silencing can also happen. In gene silencing, basically, the genes become inaccessible to the transcription machinery. So even though you have the gene in the genome, but functionally it behaves as if it is inactive. This is because the proteins which are taking part in the transcription process, they are unable to bind to the specific part of the DNA in order to transcribe it or to copy it into the mRNA. So there are certain mechanisms in which this can happen, like DNA methylation, histone modifications, and we're going to study them later in the presentation. So this kind of gene activation and gene silencing is extremely important during the development when the cells uh, obtain their individual identities. This is because the individual cells will have different genes activated while the others will be repressed by the epigenetic mechanisms. And that is how your neuron cells behave as neuron cells only, but not as the liver cells. So there is a third mechanism by which the epigenetics can control the gene regulation, and that is partial expression. So it's not always about genes simply being turned on and off. Sometimes gene expression can also be tuned. So genes can be expressed at high levels or the moderate levels or even at the low levels, which allows much more precise control of the biological phenotyping. For example, This happens in the control of hormonal levels. So most of the time, you don't simply have the inhibition of the hormones or the activation of the hormones. You have different levels of the hormones at different times of the day and also at different phases of life. So this is one way in which the epigenetics is basically regulating your genome, the genes which are responsible for those hormones at different levels. Now, these epigenetic changes are actually dynamic and also reversible, and this is one of the most important feature of the epigenetics, which means that unlike the mutations which are changing the DNA sequence for good permanently, the epigenetic marks, they change it, but these epigenetic marks can also change it back. So the changes are made in response to certain developmental signals, like some chemical signals which are present during different stages of development, or they can take place due to the environment or due to different physiologic states. But the flexibility allow the organisms to adapt their gene expression to different changing environments. Now we are going to study the mechanisms involved in epigenetic regulation. The first mechanism is DNA methylation. So DNA methylation is one of the most extensively studied epigenetic mechanism. It is quite simple. It basically involves the addition of a methyl group to the cytosine bases, which are located close to the guanine bases. So collectively we call them CPG islands, which are the hotspots for such epigenetic changes to take place. So there is addition of the methyl group to the cytosine bases, which leads to their conversion to 5-methyl cytosine. This reaction is categorized by the DNA methyltransferases, DNMT. There are certain kind of DNA methyltransferases which are responsible for different functions, like DNMT1 is responsible for maintenance of these methylation marks onto the DNA. So in many genes, such CPG islands or the clusters of CPG sites are present in the promoter region of the gene. Now the promoter region is like the entry point for the transcription machinery, because it is this area where the transcription factors will bind, and also it is this area where the RNA polymerase will bind. So RNA polymerase is the enzyme responsible for the transcription, the major enzyme responsible for that anyways. So they will bind here, but since the CPG islands are heavily methylated in the promoter region, in that scenario, the transcription factor and the RNA polymerases, they will not be able to bind here, and thus the transcription process will be halted. So when transcription will not happen, the DNA will not be read in the form of RNA, which will not be converted to any kind of protein. So this is biologically important because it helps to keep the individual cellular identity, and it also helps to regulate development. And most importantly, it helps to suppress the harmful DNA elements which are present in the human body. Like there are certain kind of repetitive DNA elements which are widespread. I mean, majority of the DNA is composed of these repetitive DNA elements only. So the DNA methylation makes sure that these repetitive elements, which basically involves the repetitive patterns of certain nucleotides like TTAG, TTCG, right? So they will be repeated on and on and on again. So DNA methylation basically keeps them repressed. And other than that, it is also responsible for the repression of something we call transposable elements. So transposable elements are basically the jumping genes which have the capability of moving about. They can move from one part of the DNA to another part. Now imagine one gene basically moving to some other part of the DNA, and that is actually that has the potential to be biologically detrimental. In animals, in plants, it is much more harmless, but in animals, it can be dangerous. So much of the transposable elements which are present in humans, they are actually repressed through the DNA methylation only, which means that they no longer have the capability of moving about. Now, this kind of mechanism is also like detrimental, especially in case of cancer. So, in cancer, there are two major groups of genes which are involved. First are the tumor suppressor gene, and the second are the oncogenes. So tumor suppressor genes are kind of like the protector genes. They are like the protective brakes in the cell cycle. They repair the DNA damage, they regulate the cell cycle, they also induce apoptosis if they consider some serious error being made in the replication process, and they also prevent uncontrolled proliferation. But in many cancers, the promoter region of the tumor suppressor genes become heavily methylated, which means that these tumor suppressor genes, they are not able to become activated. When they do not get activated, there the process of the cell cycle regulation becomes faulty, the DNA repair becomes faulty, and the cells begin to divide uncontrollably, right? The classical example of this is the mutation which is involved in the BRCA1 and the BRCA2 gene, which are also the tumor suppressor genes, and they are involved in the pathophysiology of breast cancer, also the ovarian cancer, and some other kind of cancers. Now, the second mechanism involved in epigenetic regulation is the histone modifications. Now, the histones are a special group of proteins which act as a scaffold for your DNA. So, your DNA actually wraps itself around the octamer of histones, which give rise to a structure called a nucleosome. Now, this nucleosome will be further wrapped, will be further wound, and this will give rise to a structure called chromatin, which will further give rise to a chromosome. So, chromatin is nothing but the wound configuration of DNA. And this chromatin can exist in two different configurations. It can exist in the loose configuration and the tight configuration. And this is actually regulated by some special chemical modifiers which attach themselves to the histone tails. And they basically change how tightly or how loosely the DNA will be wound around the histone proteins. The more tighter the DNA is packaged, the lesser it will be accessible for the transcription machinery, and in turn it will be repressed, or the genes will be inactivated. On the contrary, if it is loose, the genes will be accessible for the transcription machinery and the activation will happen. So the transformation, the chemical modification can happen in multiple different ways, but the important ways are the histone acetylation and the histone methylation. So histone acetylation basically involves addition of an acetyl group to the histone tails, which basically makes the chromatin more loose. So histones actually are carrying the positive charge, and your DNA carries the negative charge because of the presence of phosphate groups. So they have strong attraction for each other, and that is why they bind very closely to each other. But when acetyl groups are added, the positive charge which is present on histone proteins, it actually reduces, and because of that, the attraction between the histone and the DNA decreases, and the DNA becomes very loosely wound around the histone. And when that happens, it becomes accessible and the gene activation takes place. On the contrary, when we attach the methyl groups, this leads to the chromatin compaction, and it also leads to the transcriptional repression. However, it also depends on which protein or which amino acid we are modifying by the addition of methyl groups. So when methyl groups are added to the histone 3 protein at lysine 4 residue, then that leads to the activation. But when methyl groups are added to histone 3 protein to lysine 9 residue, that leads to histone repression. So it is actually like it depends on which amino acid residue we are methylating. Now, this kind of mechanism is essential for several different processes for maintaining the individuality of the cells. But when we discuss regarding the diseases, it has important implication in Alzheimer's disease. So in Alzheimer's, there is abnormal histone modification which contribute to the dysregulation of various genes involved in the synaptic communication, in the neuronal survival, and also the plasticity. And when some of the genes which are responsible for the healthy neuronal function becomes repressed, the inflammatory pathways, they will become overactive, and because of their overactivity, the neuronal function becomes worse and the degeneration also becomes worse. So histone modification are actually one of the most dynamic regulatory system which change the DNA regulation. The third mechanism which is involved in the epigenetic regulation is the chromatin remodeling. So this mechanism focuses on the physical organization of the chromatin. Chromatin is formed by the folding of the nucleosomes, and these nucleosomes are not fixed in their position. So these nucleosomes, they keep on changing their position with the help of special protein complexes called chromatin remodeling complexes. Many of these complexes are ATP dependent. They require ATP for their functioning. So they're also considered as ATP dependent chromatin remodeling complexes. Now, these protein complexes help to slide nucleosomes along the DNA, and they can also temporarily remove the nucleosomes altogether or reposition them, which lead to temporary changes in the architecture of the chromatin. And because of that, sometimes the nucleosomes can block important regions of the DNA called the promoter region and also the enhancer region, which are necessary for the transcription to take place. If these regions are not accessible for the transcription machinery, the transcription process will not go on, and ultimately the genes will remain repressed. So this kind of process is functionally important for the embryonic development, for the stem cell differentiation, and also for the for learning, for memory, and other kind of cellular adaptations. But with regard to the diseases, it has important implication in major depressive disorder. So in major depressive disorder, studies have found important chromatin remodeling changes which lead to changes in the chromatin accessibility and which further leads to the mood regulation and emotional processing, and basically they actually change the expression of genes which are involved in the neurotransmission and neuroplasticity and the signaling of important hormones, the cortisol, and for neuronal survival. So the altered chromatin state can lead to the lasting biological effects of stress related disorders. Mechanism involved in epigenetic regulation is the presence of known coding RNAs. For a long time, we believed that the only purpose of RNA was to serve as a messenger for carrying the information from DNA to produce proteins. But now we have shown that a large portion of genome produces the RNAs that do not code for any proteins at all, and we call them known coding RNAs for the same purpose. Now, although they are not translated into proteins, but they play a major role in regulating the gene expression. A major group of known coding RNAs involves the microRNAs. MicroRNAs are very short RNA molecules, usually around 20 to 22 nucleotides long, and they regulate gene expression mainly after the transcription has happened. So after transcription, you will have an mRNA molecule. As you can see in the diagram, there is this target mRNA molecule. The microRNA molecules, the small molecules, they will go and bind themselves to the mRNA molecule. And as they do that, they block the translation of the mRNA molecule, or they may even lead to the degradation of the mRNA molecule. Now, because of that, microRNAs are involved in important processes like regulation of the plasticity and brain development, and also the differentiation of individual cells. Another important group of the non-coding RNAs is the long non-coding RNAs. Now, these long non-coding RNAs are variable in their mechanism of action. So these are much larger RNA molecules, much larger than the microRNAs, and they regulate gene expression either by interacting with what we call the chromatin remodeling complexes, as we discussed in the previous slide. Now, these are involved in the changing in three-dimensional conformation of the chromatin. Because of that, the DNA can become more accessible or less accessible for the transcription machinery, and in this way, they can activate or silence certain genes. Non-coding RNAs can also recruit certain histone modifying, sorry for the spellings, enzymes like the HATs, histone acetyltransferases, or HMTs, histone methyltransferases. So by recruiting them, they can either make gene more active or repressed. And in this way, depending on the context, both of them can either activate certain genes or silence certain genes. Now, biologically, this process is important because it has important implication in tumorigenesis or in cancer. There are some microRNAs which can behave as tumor suppressors because they bind, they lead to the inactivation of the messenger RNAs which are coding from the oncogenes, right? So alternatively, they can also, some of these microRNAs can also serve as the oncogenes if they are leading to the degradation of the mRNA which is coming from the tumor suppressor genes. So in this way, they can basically alter the functions of tumor suppressor or oncogenes, and themselves can lead to the uh, activation of the cancer cells or the repression of the cancer mechanisms in any individual cell. Other than that, these mechanisms also have important implications in Alzheimer's disease, leads to alteration of the gene methylation and can influence your resilience to the stressors present in your environment. Another factor may involve diet, which involves the epigenetic regulation of metabolic genes and thus can contribute to metabolic diseases like obesity or diabetes. And third is the presence of certain toxins in the environment, which can disrupt the normal epigenetic activation or repression. And then there is also the involvement of early life experiences, which can program your stress response on a long-term basis and can make a person more susceptible to stress-related disorders. Now, these influences do not change the sequence. They are simply changing the epigenetic mechanisms. They are changing the normal patterns of DNA methylation, histone modifications, and chromatin remodeling. And because of that, these experiences can leave long-term molecular imprints on the gene regulation. And this has completely changed our understanding of how genetics influence us. So there was this initial debate which was going on between those who believed that genes were encoding for everything, and then there were those who believed that we are born with what we call tabula rasa, which basically means the blank slate, and we acquire every inclination after the birth. Now, with the advent of the environment epigenome interaction, we can now say that it lies somewhere in the middle. So we are not completely at the mercy of our genes, and we are also not completely independent, so that we can overcome some of the faults of our genes. we will explore one of the most fascinating and also one of the most debated questions in epigenetics, which is, can environmentally influenced epigenetic changes be inherited across different generations? So traditionally, we have always thought of inheritance as through the inheritance of DNA sequences, and that is true. But recent studies have shown that there could be another mechanism by which the inheritance could be done: extra DNA inheritance, or the inheritance of epigenetic marks. Now, before we discuss that, I want to ensure you that the epigenetic tags are actually reprogrammed or erased during the production of sperms or the egg cells. So the epigenetic tags which you are having in your own body will not be passed on to the gametes which you are producing, and thus they will not be passed on to your offsprings. For majority of the genes and for majority of the epigenetic tags, this holds true. But then there are some exceptions. There is this process called genomic imprinting, which basically means that there are certain genes which are inherited on the parent of origin basis, which means that there are certain genes which will be activated when they are coming from the father's side, and then there are certain genes which will be activated when they are coming from the mother side. So because the zygote is formed by the fusion of ovum and sperm, every individual has two copies of genes, one that is coming from the father and one that is coming from the mother. So let's say you're having a particular gene and you have one copy from the father and one copy from the mother. So genomic imprinting will decide that which gene will be inherited from which parent. So sometimes only one copy will be active while the other copy will be made inactive through the epigenetic changes, and this can be done like from on the different parent of origin basis, like the copies which are coming from fathers can be inhibited, and in case of other genes the copies which are coming from mothers can be inhibited. This phenomenon is called genomic imprinting, and this alone ensures that the epigenetic information can sometimes survive the reprogramming, because how does the genome itself know which gene is coming from father and which gene is coming from mother? Another kind of evidence can come from the sperm mediated inheritance. So researchers have discovered small non coding RNAs which can be transmitted to the zygote during the fertilization, along with the other the genetic material in the sperm. So sperm does not just inject its DNA, it also gives some of its microRNAs and other small non-coding RNAs to the zygote, which can influence the early embryonic gene expression. So studies have shown that the toxin exposure can actually change the RNA profile of the sperm, which can further lead to changes in the stress responses and the metabolic traits of the offsprings. So this also suggests that the environmental information can sometimes be transmitted biologically through the regulatory mechanisms. The strongest evidence and also the most interesting for the inheritance of the epigenetic changes comes from the transgenerational studies. So there were certain studies conducted on the humans, and these studies are basically coming from the Femine studies. And one of the most important studies was conducted during the Dutch hunger winter during the World War II, in which the Dutch populations were exposed to extreme form of starvation. So over the years, the researchers found that the children which were conceived to the women who were exposed to such conditions, they showed increased risk of metabolic conditions like the obesity and the cardiovascular condition. This is because their body is kind of expecting starvation even though they are having enough food in their own generation. So because the body was expecting such condition, it stored more of the fat than it needed, which increased their susceptibility to these kind of conditions. Similar studies were conducted in other parts of the Europe, like there was this study conducted in Sweden, which basically linked the food availability during the grandparents' development time to the health outcomes of their descendants 2 generations down or 3 generations down. So these observations suggest that the environmental stressors like famine can sometimes produce the long-term biological effects that persist across several generations. And this kind of inheritance is called the transgenerational inheritance. Now, caution must be applied when discussing these kind of findings because majority of the epigenetic marks are still being reprogrammed. Only a small amount of these epigenetic changes are escaping this reprogramming and thus are transmitted to the next forthcoming generations. And also because when conducting these studies, it's really hard to separate the effect of true epigenetic inheritance from the shared environment and also from the behavior, from the nutrition, and also from the social condition. For example, in case of the Dutch hunger winter studies, the future generations were having increased tendency of metabolic conditions. Could this be due to the epigenetic tags, or could this be because their parents and their grandparents were basically making more and more food available because of the kind of conditions that they had faced in their own life, right? So these kind of effects are difficult to separate, and thus it is hard to say whether there is true transgenerational epigenetic changes happening or whether there is just the social conditioning or the behaviors. So the important idea is this, that inheritance mostly involves the DNA sequence itself. In certain situations, the regulatory signals can get passed and we need to conduct more research to understand it further. Throughout the presentation, we have explored the central idea in modern biology, which is that the genome is not a static set of instruction. It is more modifiable, and it is modified by several epigenetic mechanisms: DNA methylation, histone modifications, and chromatin remodeling. And these mechanisms are essential for the embryonic development, for the cell differentiation, and for maintenance of individual cellular identity. So one theme through the presentation has been adaptability. So epigenetic mechanisms allows the cells to adapt to different developmental state, to different environmental condition, to different physiologic demands which are present in that individual at that particular time. Now, these experiences can leave the molecular imprints like the environmental exposures, like chronic stress or early life experiences, can influence the long-term patterns of gene expression. And in this way, the epigenome can function partly as the biological memory system. And in certain cases, these can also become dysregulated, leading to the certain conditions like the cancer. So tumor suppressor genes may become silenced in cancer, or the oncogenes may become overactive, or the stress response systems may become persistently altered, or the developmental programs can become disrupted. So the final gist of the discussion is that the genes provide the underlying code, but the epigenetic mechanisms determine when those genes will be used, where those genes will be used, and how strongly those genes will be used. So this kind of difference in the gene expression decides whether a person will be healthy or they will be deceased. Thank you.
Thank you, Sonampreet, for that interesting and insightful lecture. Taking the discussion further, we now welcome Jasmine Steffi A, who is currently pursuing an MTech in biotechnology. Her lecture is focused on understanding disease biology, connecting molecular mechanism across human diseases. Welcome, Jasmine.
Good morning, everyone. Today I'll be presenting on the topic of understanding disease biology, connecting molecular mechanism across human diseases. When we study diseases individually, cancer seems very different from diabetes, and neurodegenerative disorders appear unrelated to autoimmune diseases. However, modern molecular biology has revealed some fascinating facts. Most of them seemingly unrelated diseases are driven by common cellular and molecular disturbances. This presentation focuses on how disruption in cellular homeostasis initiate disease and how mechanisms such as oxidative stress, chronic inflammation, and mitochondrial dysfunction, protein misfolding, immune dysregulation, and genomic instability in some cases acts as a universal drivers across multiple disorders. Rather than looking only at the symptoms or affected organs, disease biology asks a deeper question. What molecular pathway has failed? And how did normal physiology become pathological, and how can this understanding help us design better therapies? By the end of this presentation, we'll be understanding how modern medicine is moving from symptom based treatment towards mechanism based precision medicine. Starting with a slide, why to study disease biology. Human disease are traditionally classified according to organs or systems. For example, Alzheimer's disease affects the brain, diabetes affects metabolism, and cancer involves uncontrolled cell growth. But molecular medicine has shown that many diseases actually share overlapping biological mechanisms. For example, oxidative stress occurs in cancer, diabetes, cardiovascular disorders, and neurodegeneration. Chronic inflammation contributes to obesity, rheumatoid arthritis, and tumor progression. Mitochondrial dysfunction is observed in aging, Parkinson's, and several metabolic syndromes. Understanding these shared pathway is extremely important because it changes how we diagnose and treat diseases. First, it enables better diagnosis through biomolecular markers. And second, therapies can target root molecular causes instead of only treating the symptoms. Third, drug repurposing becomes very possible. A drug developed for one disease may work in another disease sharing the same pathway. Finally, Finally, this forms the foundation of precision medicine, where treatment is tailored according to the patient's molecular profile. Thus, disease biology provides a unified framework for understanding human pathology. The entire concept of disease biology is that disease begins when a normal cellular homeostasis is disrupted. Cells are not static structures, as we all know. They are constantly maintained balance through tightly regulated systems controlling energy production, protein folding, DNA repair, immune signaling, and cell division. When these systems functions properly, cells survive and tissue remains healthy. However, persistent stress, mutations, environmental factors, infections, and aging can overwhelm these regulatory mechanisms. Initially, the damage may be microscopic or even reversible, but over the time, the disturbance accumulate and amplify. For example, excessive reactive oxygen species damage DNA and protein. DNA mutation alter signaling pathways, followed by misfolded proteins accumulate and become toxic, causing chronic inflammation, which again destroys the tissues. Eventually, these disturbances progress from molecular damage to cellular dysfunction, followed with tissue injury, and finally organ failure. Therefore, disease is not usually a sudden event. It is a gradual biological process driven by cumulative molecular failures. So, coming with what is normal cellular homeostasis. Before we understand a disease, we need to understand health. Cellular homeostasis is the cell's dynamic ability to maintain internal balance despite constant external stress. It's actively managed through five key systems like energy balance, where AMPK and mTOR regulate ATP production, and DNA integrity that is maintained by multiple repair systems fixing thousands of lesions per day. Protein quality control, where chaperones like HSP70 guide contract, correct and contract folding. Controlled cell division governed by cyclins and checkpoint kinases, and the different immune regulations which distinguishes a self from non-self. When one of these systems breaks down persistently, the chronic stress, environmental damage, or inherited effects, the cell begins accumulating damage and eventually targeting over different diseases. Shared mechanism across diseases. This is one of the wow moment in disease biology, I would say, because it is it provides profound insights in modern medicine with the clinical that are clinically different, because these diseases arises from same molecular disturbances. The table on this slide, maps six key mechanisms to their associated diseases. Oxidative stress drives cancer, diabetes, Alzheimer's, and cardiovascular diseases. And chronic inflammation underlies autoimmune disease, obesity, cancer, mitochondrial dysfunction links to Parkinson's, Alzheimer's, and metabolic disorders. Uh, in the same way, protein aggregation connects the major neurodegenerative diseases, and DNA damage mutation fuels cancer and premature aging, and immune dysregulation drives rheumatoid arthritis, lupus, and type 1 diabetes as well. And these overlaps are not coincidental. That's very important because they reflect the fine way a cell can fail, and that's a good news because one drug targeting a shared pathway can treat multiple diseases. Then, following with what is oxidative stress and reactive oxygen species. Reactive oxygen species are like chemically reactive molecules derived from molecular oxygen. These include superoxide anion, hydrogen peroxide, and the highly destructive hydroxyl radical. They are actually natural byproducts of aerobic metabolism, particularly from mitochondrial electron transport chain. But the accumulation beyond the cell's antioxidant capacity drives a state known as oxidative stress. Um, but the primary sources of reactive oxygen species are from mitochondria, from inflammation, sometimes from environmental toxins as well, and the radiations like ionizing and UV radiations directly cleave water molecules to generate hydroxyl ion. The molecular consequences of the excess ROS, that's reactive oxygen species, firstly it creates DNA damage, lipid peroxidation, and protein oxidation and cell death. These four can club together and cause chronic inflammation, which again causes cell damage, tissue injury, and then directly causing an organ disease. Chronic inflammation as disease driver. Inflammation is actually meant to be protective, response to injury or infection that resolves once the threat is gone. This is acute inflammation and it's very essential for the body's normal immune system. And the problem is only the chronic inflammation when the inflammatory signal persists for months or years without a real threat. At that point, it becomes destructive. Key plays includes TNF alpha, IL-6, IL-1 beta, and the master transcription factors like NF beta. Together, they drive fibrosis, genomic instability, and angiogenesis. You can see the consequences across multiple diseases in rheumatoid arthritis. The TNF alpha destroys the joints, in case of cancer, it creates a microenvironment that favors tumor growth. In obesity, the adipose tissue macrophages secrete cytokines that causes insulin resistance, and in atherosclerosis inflammatory form cells build up dangerous plaques, and then this chronic inflammation is a unifying thread through nearly all major chronic diseases. Mitochondrial dysfunction in diseases is often called the cells. These mitochondria are often called as the cell's powerhouse, but their role is far more complex than that because they regulate the apoptosis, manage ROS balance, and serves as a metabolic hubs. When they fail, through DNA mutations, impaired mitophagy, or toxin exposure. The consequences are far-reaching. Reduced ATP output stars energy-intensive neurons and heart cells. Dysfunctional electron transport chain leaks superoxide, amplifying oxidative stress. And when the damage is beyond repair, mitochondria releases cytochrome C that severely triggers cell death. In Parkinson's disease, defective mitophagy causes dysfunctional mitochondria to pile up in dopaminergic neurons. In Alzheimer's, the beta amyloid directly impairs mitochondrial complexes, and in metabolic disorders like type 2 diabetes, defects in mitochondrial biogenesis in skeletal muscle contribute directly to insulin resistance. Mitochondrial health is central for any cell survival. Followed with protein folding and aggregation. Protein must fold into its precise 3D shapes to meet its function, and the cells invest heavily in ensuring this happens correctly using molecular chaperones, the proteosomes, and the autophagy. But when these systems are overwhelmed, misfolded proteins escape clearance, clump together, and become toxic. In Alzheimer's, the beta amyloid and tau form plaques and tangles. In Parkinson's, alpha nucleosin accumulates in Lewy bodies. In different other neurological diseases, these are actually seen. And even type 2 diabetes allows IAPP aggregation in pancreatic beta cells. The most dangerous form appears to be soluble oligomers, early aggregates that disturbs membrane integrity and mitochondria. What's especially alarming is the prion-like spread of misfolded proteins. They template the misfolding of healthy protein and propagate transsynaptically, explaining why neurodegeneration follows such predictable and staggering pattern. Our DNA faces constant attack from our own metabolisms, generating loss from UV radiation, from chemicals, and from replicating errors. The cells deploy four major repair systems to handle this: basic excision repair for oxidative lesions, nucleotide excision repair for UV-induced damage, mismatch repair for replication errors, and homologous recombination for double-strand breaks. When these systems fail, the results are serious. MMR deficiency leads to microsatellite instability, a hallmark of Lynch syndrome cancer. BRCA1 2 mutations ablate homologous recombination, causing breast and ovarian cancer. Predisposition, unpaired DNA damage produces oncogenic mutation, chromosomal translocation, and aneuploidy, the raw material for cancer. This is why DNA repair pathways are now major targets for cancer therapy, including PARP inhibitors. Immune dysregulation in human disease is also very important because the immune system walks as a razor edge because it must be vigilant against pathogens and transform cells, yet it should be tolerant of its own tissues. When that balance breaks, we see immune dysregulations in two different directions. An overreactive immune response drives autoimmune diseases like lupus, rheumatoid arthritis, multiple sclerosis, and type 1 diabetes, where the immune system attacks itself. Weakened response creates immunodeficiency, enabling infections to proliferate and tumor to evade and surveillance. In lupus, autoantibodies target nuclear components, causing multiple organ inflammation. In rheumatoid arthritis, autoreactive T cells and TNF alpha destroy cartilage and bones. In cancer, tumors actively suppress immune surveillance, downregulating antigen representation, expression of PDL1, and recruiting regulatory T cells. Understanding of these regulatory mechanisms has given us the way to get through immunotherapies, one of the most significant advances in the modern medicine that can be found today. Metabolic dysregulation. Metabolic dysregulation underpins all the life, but when it goes wrong, the consequences are pervasive. Three core disruptions drive metabolic diseases: altered glucose metabolism where the tissue fails to respond insulin signal, lipid accumulation where fat deposits in organs that shouldn't store it, causing lipotoxicity, and insulin resistance where cells require increasingly higher insulin levels to function. This cascade actually underpins diabetes mellitus, obesity, and non-alcoholic fatty liver disorder. And there's a fascinating twist in cancer, that's a Warburg effect, where cancer cells preferentially uses glycolysis when in the presence of oxygen. It seems inefficient, but it actually serves cancer well, providing biosynthetic building blocks, rapid energy, and creating an acidic microenvironment that promotes invasion. So, when a metabolism is co-opted by malignancy, the connection between metabolic diseases and cancer are deep and bidirectional. Cell signaling pathways, like every cellular response towards environment depends on the signaling pathway. Molecular relay system that transmit instructions from outside the cells to the nucleus. When these pathways are disrupted, cells receive the wrong message. The NF factor, the master inflammatory switch, is consecutively active in many cancer and inflammatory diseases. The PI3K AKT axis, central to growth and survival, is hyperactivated in numerous tumor types, driving uncontrolled proliferation. The ERK and P38 promote proliferation in cancer and inflammation in autoimmune conditions. The JAK-STAT pathway, which carries cytokine signal, drive autoimmune disease and hematologic malignancy. When aberrantly activated, the great therapeutic opportunity here is clear. Target these pathways with small molecule inhibitors and monoclonal antibodies. The challenge is that tumors evolve compensatory roles, which is why combination strategies are increasingly essential. Followed with neurodegenerative diseases and its molecular mechanisms. Parkinson's disease offers a powerful case study in how multiple molecular mechanisms converge. Affecting 1% of the people over 60, it is characterized by the loss of dopaminergic neurons in the substantia nigra. Here's what that is happening at a molecular level. First, alpha nucleosin misfolds and forms Lewi's body, disrupting the proteostasis and spreading between neurons. Second, dopamine metabolism itself generates ROS, making them these neurons especially vulnerable to oxidative damage. Third, pink protein, that's pink1 and Parkins protein mutated in familial Parkinson's, fail to clear damaged mitochondria, allowing dysfunction to accumulate. And fourth, activated microglia create chronic neuroinflammation that amplifies neuronal damage. Notice how all four mechanisms reinforces each other. Nucleus aggregates actually impair mitochondria, then followed with dysfunctional mitochondria increasing ROS promoting misfolding, and all of it triggers inflammation. This synergistic interaction explains why single target therapies have largely failed, because combination approach targeting these interconnected networks is the future. Cancer biology and its molecular basis. Cancer is not just one disease, it is hundreds of diseases sharing a common onset of acquired capabilities that Hahn and Weinberg famously described as hallmarks of cancer. These include sustaining growth signals autonomously, evading tumor suppressors, resisting cell death, inducing angiogenesis, enabling invasion and metastasis, and evading immune destruction. On the molecular side, cancer accumulates genetic alteration, oncogene activation through mutation or amplification, tumor suppressor silencing, repair defects, and chromosomal rearrangements. Environmental factors also contribute, like chronic inflammation, oncogenic viruses like HPV, EPV, and chemical carcinogens and radiation. The good news is that understanding these hallmarks have driven rational drug development, tyrosine kinase inhibitors, PRP inhibitors, immune checkpoint blockers, and anti-angiogenic agents. The ongoing challenge is that tumor heterogeneity and evolutionary resistance making adaptive combination strategies a priority. This molecular mechanism when it comes to diabetes mellitus. Diabetes provides a clear example how different molecular insults can coverage one same clinical syndrome. In type 1, it's autoimmune, autoreactive T cells infiltrate into the pancreatic islets, recognizing the self antigens and destroying B. The result is absolutely insulin deficiency. This process can unfold silently for many years before the symptoms appear. In type 2, the problem starts differently. The insulin resistance in liver, muscle, and fat, driven by chronic inflammation, lipotoxicity, and mitochondrial dysfunction. Initially, beta cells compensate by producing more insulin. Eventually, they fail. The downstream consequences of chronic hyperglycemia affects every system through four pathways: age formation, polyol pathway activation, PKC signaling, and hexosamine pathway flux. These molecular disruptions drive the devastating complication of the disease: neuropathy, nephropathy, and cardiovascular disease and retinopathy. Managing diabetes well means managing all these molecular cascades, not just blood sugar. The interconnection between different diseases. Yeah, by a pattern that should be clear now. Like every disease is not an isolated event, they are interconnected nodes in a molecular network. The same mechanism that drives metabolic disease also fuels neurodegeneration, cancer, and autoimmunity. This isn't just academically interesting, it has real therapeutic implications. Metformin and diabetes drugs shows anti-aging and anti-cancer effects. Anti-inflammatory therapies shows benefits across multiple disease categories like lifestyle interventions like exercise, calorie restrictions, improve mitochondrial function and reduces inflammation and hence stress resilience all at once. The slide illustrates a practical cascade. Obesity drives adipose tissue inflammation, which causes insulin resistance, which precipitates type 2 diabetes and the cardiovascular diseases all through shared molecular mechanism. The system biology approach that maps these disease networks helps us to identify the clinical and critical points for multi-targeted intervention. The future of medicine is network pharmacology, not single target treatments. The future is precision medicine. Precision medicine represents the translation of everything we have discussed into a personalized patient care. It begins with genomics, sequencing a patient's genome to identify disease causing mutations, pharmacogenomic variants. It adds biomarkers, a measurable indicator like circulating tumor DNA that enable early detection and treatment monitoring. And it also builds a molecular profiling integrating transcriptomics, proteomics, and metabolomics to create a comprehensive disease signature. The results are striking. In biomarker selected cancer patients, response rate improves from 10 to 20% with the standard chemotherapy to 50 to 80%, which is huge with this targeted therapies. EGFR inhibitors for lung cancer, BRAF inhibitors for melanoma, PRP inhibitors for BRCA mutant cancer, and cystic fibrosis, once uniformly devastating, is now manageable with CFTR modulators tailored to specific mutations. CRISPR gene editing is opening doors to curing monogenic diseases. We are in the early days of this revolution, but the trajectory is wide open and clear. Emerging technologies that are used in revolutionizing disease biology research. The phase of discovery biology is accelerating, driven by five transformative technologies: AI and machine learning are diagnosing diseases from imaging to with superhuman accuracy and designing novel therapeutics. CRISPR gene editing enables precision genome modification for research, for disease modeling, and increasingly for therapy. Single cell sequencing reveals the heterogeneity within tissue that makes that bulk analysis mask, identifying rare cell population and mapping tumor microenvironments at unprecedented resolution. Multi-omics integration combining genomics, transcriptomics, proteomics, and metabolomics. This reveals a regulatory network invisible to any single approach. And organoids, patient-derived 3D tissue model allows us to test the drugs in human-relevant systems before animal studies. The true power comes from integrating these technologies. CRISPR screens in organoids guided by AI analysis for single-cell multiomics data. This convergence is almost expressing the timeline from basic discovery to clinical translation. For conclusion, I would like to bring you all of your attention. The central insight of this webinar is that diverse diseases share common molecular mechanisms like oxidative stress, chronic inflammation, mitochondrial dysfunction, protein aggregation, DNA damage, and signaling dysregulations. These processes create self-amplifying cycles like oxidative stress damages mitochondria. Dysfunctional mitochondria produces ROS, which triggers inflammation, which further increases oxidative stress. Breaking this cycle requires multi-targeted system-level approaches. Precision medicine is powered by genomics, biomarkers, and emerging technologies, gives us the tool to exactly do that. Looking ahead, within a decade, we anticipate routine genome sequencing at birth, probably liquid biopsies for early cancer detections, and gene editing cure for inherited disorders. The final thought I would like to leave here is understanding disease at a molecular level transforms medicine from reactive symptom management to proactive health optimization. We are moving towards preventing disease before it starts, targeting its root cause, not downstream effects. Thank you so much for joining with me today.
Thank you, Jasmine, for delivering such a meaningful and broader perspectives on disease biology. Our final lecture for Theme 5 will be delivered by Aditi Dubey, who comes from a background in applied microbiology and biotechnology. Her lecture will be focusing on cancer as an evolutionary disease. Welcome, Aditi.
Good morning, everyone. I am Aditi Dubey, and today I'll be speaking on a topic that has fundamentally transformed our understanding of oncology: cancer as an evolutionary disease. Traditionally, cancer was viewed simply as uncontrolled cell growth caused by mutations. However, modern research has revealed that cancer is much more dynamic. Tumors are not static masses of identical cells. Instead, they are evolving ecosystems composed of genetically diverse populations of cells that continuously adapt to their environment. Much like organisms evolve in nature through Darwinian selection, cancer cells evolve within the human body through mutation, competition, and survival under pressure. Understanding cancer through the evolutionary lens helps explain one of the biggest clinical challenges in oncology: why treatments initially work but later fails. Today, I will discuss how tumors evolve, how heterogeneity develops, and why resistance emerges, and how this knowledge is shaping future therapies. So now let's look into why the traditional view is incomplete. For many years, cancer was described using a linear model. A normal cell accumulates mutations, becomes malignant, forms a tumor, and spreads. While this model is useful, it is incomplete. The modern view sees cancer as a population of millions or billions of cells, each potentially different from others. Within a tumor, multiple subpopulations or clones compete for nutrients, oxygen, space, and survival. Some clones grow faster, some evade the immune system better, but survive therapy. So rather than a single disease entity, cancer is more accurately a constantly changing ecosystem. This shift from a static model to a dynamic evolutionary model is one of the most important advances in cancer biology. To understand this concept, let us connect cancer with Darwinian evolution. In natural evolution, three components are essential: variation, selection, and inheritance. The same occurs in tumors. Variation. Cancer cells acquire mutations due to DNA damage, replication errors, or genomic instability. Then comes selection. The tumor environment, immune system, low oxygen conditions, and therapies create pressures that eliminate weaker cells. And then inheritance. Cells that survive, divide, and pass advantageous traits to daughter cells. Over time, fitter clone dominates. This means cancer progression is not random chaos. It is an evolutionary process driven by selection. So now let's look into what is clonal selection. Clonal selection begins when one normal cell acquires key driver mutations that allow uncontrolled growth. That cell divides repeatedly and forms a clone, a population descended from original transformed cells. As the clone expands, additional mutations arise. Some daughter cells gain new advantages, that is, faster growth, resistance to apoptosis, immune escape, and drug resistance. These successful descendants expand further, creating new subclones. Thus, tumor evolves through branching patterns, not straight lines. This model is called clonal evolution, first proposed by Peter Nobel in 1976 and remains foundational today. So, not every mutation contributes to cancer. Tumor genomes often contain thousands of mutations, but only a subset actively drives progression. So, what is driver mutations? These provide survival or growth advantages. Examples: TP53, KRAS, EGFR. Now, passenger mutations. These occur incidentally and do not contribute directly. Understanding the difference is crucial because modern targeted therapies are designed against driver mutations. However, tumors can evolve around these therapies by generating new drivers. Now let's move to heterogeneity. One of the most important consequences of evolution is heterogeneity. This means cells inside the same tumor can differ dramatically. Within one tumor, cells may vary in mutation profile, gene expression, metabolism, growth rate, ability to metastasize, drug sensitivity. This is called intra-tumor heterogeneity. Even two neighboring cells may behave differently. This diversity acts like biological insurance for the tumor. If one treatment kills some cells, others may survive. That is why eliminating all cancer cells is so difficult. As we are talking, biopsies sample only a small part of tumor. That means the sample may miss dangerous resistant clones elsewhere. The treatment response becomes unpredictable. Some cells may die rapidly while others persist. Then heterogeneity increases reoccurrence risk. After treatment, surviving clones can repopulate the tumor. And finally, metastatic lesions may differ from primary tumor genetically. That means treatment chosen from the original biopsy may no longer be optimal. Therefore, heterogeneity is not just a biological curiosity. It directly affects patients' outcome. So after heterogeneity, let's look into drug resistance. Drug resistance is perhaps the clearest example of tumor evolution. Imagine a tumor with 99% sensitive cells and 1% resistant cells. So when the therapy begins, sensitive cells die, resistant cells survive, competition is removed, and resistant cells expand. Eventually, the tumor returns, now dominated by resistant clones. This resembles antibiotic resistance in bacteria. The treatment did not create resistance from nothing. Often, resistant cells already existed in small numbers and were selected. Thus, therapy itself can unintentionally shape tumor evolution. So now let's look into the types of drug resistance. There are three main forms of resistance: intrinsic, acquired, and adaptive. In intrinsic resistance, the tumor is resistant from the start due to the pre-existing mechanisms. While in acquired resistance, initially sensitive tumors later evolve resistance after treatment exposure. And in adaptive resistance, cells temporarily reprogram themselves under stress without permanent mutations. Examples include activation of alternative pathways, dormancy states, or epigenetic changes. This shows resistance can arise through both genetic and non-genetic evolution. Now let's go through one example: EGFR mutant lung cancer. A classic example comes from non-small cell lung cancer. Some patients carry activating mutations in EGFR. They often respond dramatically to EGFR inhibitors. Tumors shrink, symptoms improve, and progression slows. However, many patients later relapse. A common cause is the emergence of T790M mutation, which alters the drug binding site. This resistant clone expands under treatment pressure. Newer drugs were then developed specifically to target T790M. This cycle beautifully illustrates co-evolution between medicine and cancer. Now let's go through tumor microenvironment as selective pressure. Cancer cells do not evolve alone. They interact with the tumor microenvironment, including immune cells, fibroblasts, blood vessels, extracellular matrix, nutrient gradients, and low oxygen regions. These factors create selective pressures. For example, low oxygen may favor cells that switch metabolism. Immune attack favors cells that hide antigens. Dense tissue favors invasive clones. Thus, evolution is shaped not only by mutations, but also by ecological context. Now let's look into the cancer development process. Metastasis is the spread of cancer to distant organs and is responsible for most cancer deaths. But metastasis is inefficient. A cancer cell must detach from the primary tumor. So, how do we track tumor evolution today? Modern technologies now allow us to observe evolution in real time. Single cell sequencing, liquid biopsy, ctDNA monitoring, and artificial intelligence are helping us to track evolution. Let's start with single cell sequencing. It measures mutations and gene expression cell by cell. In liquid biopsy, it detects tumor DNA fragments in blood, while in ctDNA monitoring, it tracks emerging resistance mutation before scans show relapse. And AI, the artificial intelligence, analyzes the large data sets to predict progression and treatment response. These tools are shifting oncology from reactive care to proactive evolutionary management. From the beginning of this lecture, we are seeing that cancer is continuously evolving. So if the cancer evolves, treatment must also evolve, right? So now let's look into new therapeutic strategies. Let's start with combination therapy. In combination therapy, it attacks multiple pathways simultaneously to reduce escape routes. Then comes the sequential therapy. It uses changing strategies over time based on tumor adaptation. Then there comes the adaptive therapy. Instead of maximum killing, maintain a stable sensitive population that suppresses resistant clones. And finally, precision oncology. Tailored treatment based on each tumor molecular profile. These approaches aim not just to destroy tumors, but to steer their evolution. Now, let's look into the future perspective. Cancer as a chronic ecosystem. The future may treat cancer more like chronic ecological management than total warfare. Rather than trying to eliminate every cell immediately, which often selects resistance, we may control populations strategically over time. This could convert some cancers into manageable long-term diseases. The key message is simple. To defeat cancer, we must predict how it adapts. So now let's summarize the whole long lecture. There are some key takeaways. Starting with, cancer evolves through mutation and natural selection. Tumor contains diverse competing clones. Heterogeneity drives relapse and metastasis. Drug resistance is evolution under therapy pressure. And finally, future oncology must become adaptive, personalized, and evolutionary. So, as I conclude, I would just say cancer is not just a genetic disease, it is an evolving biological system. The more deeply we understand its rules of adaptation, the better we can design therapies that stay one step ahead. Thank you everyone for listening to this whole lecture patiently. I hope you all liked it. Thank you for your attention.
Thank you, Aditi. We now move into theme 6, drug discovery fundamentals, from biological targets to therapeutic design. This session will focus on how biological understanding is translated into therapeutic strategies, covering target identification, drug development pathways, computational approaches, and modern therapeutic design. Our first lecture in this session will be delivered by Poshali Bose, who comes from a background in biophysics, molecular biology, and bioinformatics. Welcome, Poshali.
Hello, please let me know if I am audible. Please, am I audible?
Yes, you're audible.
Okay, thank you.
Poshali, are you sharing your screen? Because
Yeah,
we
sharing my
can't
screen.
see that.
Can you see my screen? Please let me know if you can see my screen.
Yeah, we can.
Okay, thank you. So, hello everybody. My name is Paishali Ghosh. I'm also coming from drug discovery background. So,
Sorry to interrupt.
yeah,
Some issue with the volume. I think I can, like you can speak a little louder.
okay, okay. Am I audible properly now?
Yeah, perfect.
Okay. So good afternoon, everybody. So today I'm going to talk about drug discovery fundamentals from biological target to therapeutic design. So my name is Paushali Bose. I'm also coming from the drug discovery background. So what I'm going to talk today is that I'm aiming to provide a structured overview of how we actually translate the biological understanding into therapeutic interventions. while also highlighting emerging advancement shaping the future of this field. So, before going into the details, before we digging into it, I would like to talk about what are the topics that I'm going to cover today. So, why drug discovery matters, then giving a brief introduction about the disease overview, and then how can we actually find a new drug target, followed by the target identification. We need to validate the target because sometimes there are a lot of targets are not druggable, so that needs to be validated as well. After that, we'll dig into the lead discovery and lead optimization procedure, followed by preclinical testing, clinical trials, and an example of the case study, and then how AI is actually evolving in this field, and then a brief summary of it. So before going into it, we'll talk about why drug discovery matters. So at this outset, we know it's very important to reflect on why drug discovery remains critically important. So despite remarkable progress in biomedical science, a significant proportion of disease still lack effective or target therapies. So it includes different complicated conditions, for example, cancer, neurodegenerative disorders, and rare genetic disease. So here from the slide, we can clearly see that a lot of diseases whose medical needs are still unmet because the clinical reasons are still not known. Other than that, there are a lot of other diseases whose drug resistance have already been developed. For example, TB is one of those disease which requires urgent discovery. Otherwise, it can be a global threat by 2050. Other than that, there are a lot of diseases, especially like cancers or metabolic disorders, especially where the patient, where the system varies patient to patient. So every patient needs different individual treatment or personal approach. So drug discovery is very relevant in that aspect as well. So at the end of the day, we know that in developing country, the number of cancer patients and other diseases, the way it's increasing at faster rate, it's very harder for maintaining the doctor and patient ratio. It's very essential to manage this thing in such a way so that this healthcare burden can be reduced and the individuals can get the proper treatment. So it also new scientific innovations, and at the end of the day, which causes the economic growth of the country. So basically, it can help us to prepare for even better future, and to avoid not to avoid the situation like happened during the COVID, but to have a better way to combat with the problems if it happens all of a sudden. So drug discovery is not merely about developing medicine, but it's about addressing the unmet clinical needs, improving patient outcomes, and advancing global healthcare systems. However, the process is inherently very challenging. It is resource intensive, time consuming, and characterized by a high rate of attrition, where only a small fraction of candidates ultimately succeed. So next we will go talk about the disease. I know at this point everybody knows what is disease, just a departure or from the ease point of view. So basically our body gives different types of symptoms because already the problem has already started happening. But we are good, for most of the time we are good at ignoring it, or sometimes it happens the symptoms are very common for different diseases. It becomes very hard for us to give attention to any particular symptoms. So the disease can be of different types. We know that it can be acute and it can be chronic as well. But apart from that, we can classify the diseases based on the source, because it's very essential to know the source of the disease or the cause of the disease in order to find the proper treatment for that. So we know there is infectious disease which can be caused by bacteria, virus, and fungi and parasites. The non-communicable diseases are mostly the cardiovascular disease. And other than that, genetic and hereditary diseases are there, which are actually carried over the generations. For example, the BRCA mutation is one of those that has been discussed over in almost every slide, every presentations before. Other than that, nutritional deficiency is also there, which can actually be cured just by supplements. And other than that, autoimmune diseases, which is actually a complicated problem, especially for the patients, like cancer patients, especially if somebody is dealing with autoimmune disease, the immunotherapy becomes very difficult for them to get treated with. Other than that, congenital birth defects are there, but which can be surgically treated as well. So, any successful drug discovery program begins with a deep understanding of the disease biology, which involves there's molecular mechanisms, cellular pathways, and specific interactions that drive disease progression. So, for example, I have already talked about the metabolic disorder here, that we know that the diabetes is caused by the insulin, but again, overdose of insulin can lead to a lot of problem because it's actually a growth hormone. So, having the medications at proper dose is also very important. And also, precise understanding of these mechanisms are very essential because it forms the foundation for identifying the actionable therapeutic strategies. So next, what is the status of the drug discovery or the strategies in modern medicine? So basically, once we know the disease, we can identify the problem. And once the problem is known, we can have two options. Either we can have the drug. If the drug is not there, we have to start from scratch. Or sometimes it happens the drug is still existing, but it has been resistant. So basically, if there is no drug, then we have to start from the very scratch, where we have to understand the disease biology first, which includes the pathway, pathogenesis, all the genetics, and biomarkers. For example, in case of SARS-CoV-2, which takes a lot of time to know that it's a spike protein which is actually causing the problem. Once we know this, we can identify the target. But this target identification is very laborious. We'll talk more about that in details later on, which involves actually a lot of informatics analysis of different kinds of omics data that actually eventually help us to get or identify new targets. And then we need the experimental validation of those data by doing the gene knockouts so that we can actually do the phenotypic screening between the wild type and the mutated gene or the knocked out gene. And then finally we can develop the new drug. So basically the drug discovery part comes in two ways. One, identify the disease. Second step is identify the target. And third step, once the target is identified, go for the identification of new molecule or find out the small molecular inhibitors or just repurpose the drug that is already existing. If we already know the drug resistance have evolved, we can first go for drug repurposing because the pharma company has a lot of molecular depository in their pharma bank. So basically, they can go for the molecular screening of those. Just by changing some of the functional groups can actually repurpose the drug. That I'm going to talk about later on how say carboplatin, which is a famous ovarian cancer drug, has just been repurposed from the cisplatin just by changing the functional group. So having a lot of library is very crucial because it gives us a better way with the help of AI to go to find out a better solution for the disease that we are finding difficult to tackle with. And another thing is new target discovery. Again, for this reason, for this purpose, we have to do very minute network analysis in order to find out what can be considered as a good target. And then combination therapy, that has already been mentioned in the previous lecture, where actually two different kind of targets are actually being tackled together so that disease can be under control. So next I'm going to talk about the drug discovery platform, drug discovery pathway. So basically this drug discovery pathway is not a very straightforward pathway. This pipeline actually involves a lot of steps, which actually is very time consuming. So here you will see the process begins with the disease understanding and target identification, followed by the validation, hit discovery, and lead optimization. So subsequently, compounds undergo preclinical testing and clinical evaluation before seeking regulatory approval. Even after the approval, the post-marketing surveillance is still conducted to ensure the long-term safety. Because of the different ethnicity can have different effects. Overall, this is highly complex and lengthy process, often spanning over a decade and requiring substantial investment, with no guarantee of success. So we'll talk about each of these stages later on. So now let's come to the point, what is actually the drug target, or how can we find out the target. So basically the next step is target identification, where potential targets are systematically identified using all these advanced bioinformatics tools. So these mainly include the genomics and transcriptomics data and proteomics data as well. And then once we use all these huge data banks, we get to know the internal network. Like for example, which gene is responsible for overexpression of the other genes. So this is how we are able to know the difference between the role of the important gene other than those housekeeping genes. Because if we hamper the housekeeping gene, a lot of other pathways can be involved. So this is very crucial step to identify the proper target. So basically, this stage often generates a broad set of candidates. However, Identifying the target is only the beginning. The real challenge lies in determining whether it can be therapeutically explored or not. As I have mentioned before, a lot of targets are there which cannot be druggable, because we know based on the chemistry, when suppose if I'm talking about an enzyme, or if an enzyme is interacting with an analyte or it's a substrate, it might form transition states. Sometimes we know the transition states does not last long, more than nanoseconds. If it lasts long enough, then it can be drugged, and if it is not, then that cannot be druggable. So we need to know it's not about just identifying the target. We have to also look at the point where it can be druggable as well or not. So mostly it's proteins, DNA, RNA, and different pathways, which are actually the target, can be good drug target. So target identification is not sufficient alone. It requires validation as well. So at this point, it is considered to be the critical decision-making stage, because here we are aiming to establish a causal relationship between the target and the disease. In other words, we ask, does modulating this target produce a meaningful therapeutic effect? This is typically achieved through both wet lab work and as well as computation approach. So basically, in silico approach, we use different multi-omics data, and then we identify the genetic variants, which will help us to know, okay, if this is the gene which causing overexpression of other genes or not, and then we can target the gene in that particular pathway to find out the solution of that problem. Other than that, we use the predictive modeling, where we can simulate the protein function, protein-protein interaction, protein-DNA interaction, even protein-RNA interaction as well. So it's not about the protein. So if we are able to block the binding of protein and protein DNA, protein RNA, that can also bring a lot of changes, because the things are not happening at genetic level. It's also happening at epigenomic level and epitranscriptomics level as well. So this is how we can actually modulate the target modification, and then prioritize the targets for druggability assessment. But other than computation approach, we definitely have to do wet lab validation, where two parts are come into play. One is genetic manipulation, where the target gene is actually knocked out by CRISPR or siRNA mediated pathway, and then the mutants and wild type will have different phenotypic effects, which will be screened, which is usually screened. And then other than that, the gene has usually puts back to its place to see the overexpression or gain of function or loss of function. These kind of studies are important in order to know the dosage of the analyte or the drug that is needed later on. Other than that, there is another stage, that is the pharmacological tools, where all these molecules that have been screened for need to be undergo through this phase, because we need to understand the interaction kinetics between the drug and the target. So we need to know the kinetics behind it, that is the on rate and the off rate of the molecule, in order to know the inhibitory concentration and as well as binding constant, which will help us to know the half-life of the molecules later on. So these steps include mostly different types of biophysical assays, for example SPR, IPC, thermal shift. All these assays are required in order to know the binding affinity of the molecule or the drug towards the target, so which is usually done on the animal models, and uh, as a part of in vivo study. So these both stages are crucial in order to do the target validation. My screen is frozen. Okay. So this is one of the crucial steps, where the target has been identified. So now once we know the target, we can go to the further next step, where we need to identify the molecule that can actually bind to the target and inhibit its function. So next stage is hit identification or the lead validation. So basically, hit or lead identification is like, once the target is validated, the focus shifts to identifying molecules that can interact with it. So this is known as hit or lead discovery. So large libraries of compounds are there, which involves high throughput screening and as well as orthogonal assay, dose dependent studies, to reduce down the number of compounds, so that it can be managed further for the lead optimization process. So lead optimization is also very crucial step, because at the end of the lead identification and validation, there is a huge list of compounds, and most of them will eventually not even work. So their thermodynamic study needs to be done, and as well as their pharmacokinetic and pharmacodynamic property needs to be done as well, which is the part of the lead optimization procedure. So this initial hits are then refined to a process known as lead optimization. This stage is highly iterative and multidisciplinary, which is involving mostly medicinal chemistry, structural biology, and pharmacology, all of this background. So the purpose of these lead optimization stage is to refine the potency, selectivity, pharmacokinetic properties, and safety profiles. So that by the end, small structural modifications can lead to significant improvements, ultimately resulting in a candidate that is suitable for further development, will have eventually uh, less attrition rate. So basically, this is mostly done by computational methods like structure activity relationship studies, where mostly once the lead is discovered, we can actually modify different groups, and then we check the admit property, which actually involves the absorption, resolution, and metabolizing and excretion property of the molecule, so that we will be able to know what is the dose required, what is the half-life of the molecule. So these kind of studies are required, which is the part of the lead optimization study. And it's mostly done by computationally through different kind of docking softwares. So before going into the next preclinical testing, I would like to briefly summarize how in each of these stages AI is playing crucial role. So basically, if we consider the beginning, where the genomic and disease foundations were, was helping us to identify the target, was actually can be done by AI or mostly the bioinformatics tool. But this omics data can help us to know if the target is druggable or not. Other than that, we know that during this drug discovery procedure, a huge compounds are actually being screened from the library, and it's very difficult and financially puts a lot of constraint. So to identify each of the molecular interaction with the target, and sometimes it's not even feasible to do because the machines like SPR and other biophysical instruments, it takes months to validate one particular method. So in that case, AI is actually helping us to generate the molecular space computationally, so that we can actually do the fast binding prediction. We can actually generate the de novo molecules as well with the help of the AI. Other than that, we know the structural biology, where we are getting the structure of the molecule, and we can just dock our molecule or the drug on the target, and can simulate them computationally, so that we will get the docking score or binding prediction. The more negativities, the more it is likely to have more effect on the inhibition of that particular compound. Other than that, as I have already mentioned, the admit profiling, where the empirical formula of the molecule or the smiley, which is actually used by the AI to do the pharmacokinetic analysis. So we are seeing how in each of these stages, computational part is helping us in order to overcome the problem. Next, I'm going to talk about the preclinical testing. So before entering the human studies, candidate drugs undergo rigorous preclinical evaluation, which includes both in vitro studies using cellular system and in vivo studies in animal models. So these key parameters are, which needs to be assessed, are toxicity, pharmacokinetics, and pharmacodynamics. The only compounds that demonstrate acceptable safety and efficacy profile progress to this clinical trial, because the target of the clinical trial is to achieve the safer drug, or the drug which is safer to all major group of people. The clinical development is conducted in three sequential phases. Phase 1 focuses on safety and dosage in a small group of healthy volunteer or patients. Phase 2 evaluates efficacy and further assess safety in a larger patient population. And phase 3 involves large-scale studies to confirm efficacy, monitor adverse effects, and compare with existing standard treatments. So it is very important to note that even at this advanced stage, future failure rates remain significantly high. And it takes at least 15 years for all these clinical trials to be done, and for a medicine to come in the market. And then I will briefly talk about a case study where I would like to talk about the carboplatin, which is actually considered to be repurposing of the cisplatin. Which cisplatin is actually a serendipitous discovery by a scientist when she was actually growing something on the plate, and she has seen that during the kind of electrochemical studies, somehow actually something has stopped the growth of the bacteria, and eventually she found out that it was actually the platinum. And we know platinum is inert metal, so it has a lot of nephrotoxicity. So that's why the fluoride group, as I mentioned, just one functional group change can lead to improved admit properties. So similarly, the fluoride group of that cisplatin has been changed to carbon to another butyl group, cyclobutyl group, which actually enhance the admit property by reducing the interaction with the other parts of the body. It still has the nephrotoxic effect, but it still has less effect in terms of the cisplatin. So that's how carboplatin has been revelation to the ovarian cancer mostly, and also as well as breast cancer. So this is an example how repurposing of drug can actually be useful if we do this admit analysis properly with the help of AI. So there are different generative models for molecular design. Mostly, as this is kind of the network models and the diffusion and language models that we generate that actually we use from the molecular structure or the smileys. So once we do that, it gives us a better representation to capture the chemistry for downstream designing the task. And other than that, this sorry, this AI is also can help us to get the and the better understanding of the admit property, which can help us to know the drug which will have better potency and selectivity. By optimizing this workflow, we can actually tackle the real world problem in a better way. So before the end of this talk, I would like to talk about the effect of the AI recently. So we can see the admin property and toxic endpoints. All these things have been improved once the AI has come in play. According to the recent literature, with the help of AI, the drug discovery field is actually increasing with the rate of 10%. So it's pretty good news that we can see that a lot of things which are actually laborious and kind of the tedious job and kind can be done with the help of AI, but which needs a better analysis of the data, which is less time consuming in comparison to doing the manual work, which can be done by the AI. Also, AI is actually all these neural network are mostly the iterative process, so helps us to get a better molecular development to tackle the problem in a better way. Thank you for listening to this.
Thank you, Pushali, for that foundational overview. Continuing our discussion on therapeutic development, we now welcome back Ashley Verghese. She will be delivering her lecture on drug discovery fundamentals from biological targets to therapeutic design. Welcome, Ashley.
Hello, everyone. I am Ashley Workers, and welcome to this session Drug Discovery Fundamentals from Biological Targets to Therapeutic Design. Before we begin, I want you to think about something very simple. Whenever we take a medicine, we expect it to work instantly. But what we often don't realize is that the small tablets is a result of 10 to 15 years of research and billions of dollars in investment and thousands of failed attempts. So the real question is, how does a drug actually go from an idea in a lab to something we can use in patients? This is exactly what we will explore in today's session. Drug discovery is the process of identifying, designing, and developing compounds that can treat diseases effectively and safely. But to understand this deeply, we need to break it into levels. Every design begins with a molecular problem, maybe a mutated gene, dysfunctional protein, or a disrupted signaling pathway. For example, in cancer, it is a uncontrolled cell division, or maybe in diabetics, it may be caused due to insulin signaling issues. So the level 2 is chemical intervention. That is, once we identify the problem, we need to design a molecule that can effectively block a harmful protein, or it can activate a missing pathway, or it may restore a balance. So third one is pharmacology or therapeutic translation. That is, we convert that molecule into drug that may reach the tissue rightly and it effectively work. That is, it causes minimal side effects. So drug discovery is not just about finding molecule, it is about understanding life at molecular level and correcting its errors. So as you listen, try to connect each step with real diseases you know. So now we are going to discuss the drug discovery pipeline. So it may include lots of process. First one is target identification. Next, the validation of the targets, that is, it confirms the target's role in disease, followed by lead discovery and lead optimization, which screens the compounds for activity and it improves the potency and safely. The next stage is preclinical testing, followed by clinical trials, and finally the approval of a regulatory committee. Now we enter the most critical step, identifying what to target. A drug target is usually a protein, enzyme, receptor, or maybe a gene. For example, in autoimmune disease, overactive cytokine like TNF alpha drive the inflammation. So drugs are designed to block this TNF alpha. Target identification methods include genomics, proteomics, as well as bioinformatics. So, once we identify the target, we need to validate it. So, this step is very crucial because it helps to ensure the target is actually meaningful. So, we use knockout models, that is, we remove the genes and observe the effects. Next is RNA interference, that is, we silence the gene or may use precise gene editing such as CRISPR-Cas. So the key concept of target identification and validation is druggability. That is, not all drugs are suitable. Some proteins lack binding pockets or may be structurally unstable or are essential for normal functions. So targeting them may cause toxicity. So a wrong target means years of research wasted. This stage determines the entire success of the pipeline. So, next is lead discovery and optimization. So, once the target is validated, we need a molecule that interacts with it. That is the lead discovery. How do we find leads? So, first method is ATH, that we test thousands of compounds rapidly. Next is the virtual screening. We use computational docking. The last step is using natural products. Many of the antibiotics comes from microbes. So from hits to lead, that is initial activity followed by optimized candidates. So lead optimization includes refining a molecule based on increased potency, improved selectivity, reduced toxicity, and enhanced stability. So leads undergo iterative chemical modification to improve these above criteria. And in order to achieve this lead optimization, star analysis is used and it guides the medicinal chemistry decision. Now that we have identified a optimized a promising drug candidate, the next step is preclinical evaluation. This stage acts as a bridge between laboratory research and human trials. In simple terms, this is where we answer one key question: is this drug safe and effective enough to be tested in humans? So, preclinical studies involves a series of experiments conducted before human testing. They are designed to evaluate safety, biological activity, and mechanism of action, as well as pharmacokinetic behavior. At this stage, most drug candidates fail, and this is actually a good thing because it prevents unsafe or ineffective drugs from reaching human. So, let us begin with in vitro studies, which are performed outside a living organism, typically using cultured cell. These studies helps us understand whether the drug interacts with the targets, its mechanism of action, and cytotoxic effects on cells. For example, cancer drugs are tested on tumor cell lines. Antimicrobial drugs are tested against bacterial culture. So, we measure parameters like cell viability, enzyme inhibition, and receptor binding. One important advantage of in vitro studies is control. We can precisely control the environment and isolate the specific effects. After the in vitro success, the drug moves into in vivo studies, which are conducted in animal models. So, commonly used animal models are mice, rats, sometimes primates. So, these studies provide insights into drug efficiency in a living system, systemic toxicity, organ specific effects. For example, Does the drug reduce the tumor size? Does it affect the liver or kidney function? So this stage is crucial because it reflects how the drug behaves in a complex biological system where multiple pathways interact simultaneously. However, there is a very important limitation. Results in animal do not always translate to human. So this is one major reason for drug failure in later stages. So so now we move on to one of the most critical aspects after in vivo and in vivo testing, that is ADME profiling. So ADME stands for absorption, distribution, metabolism, and excretion. So let us break down each term. Absorption, how the drug enters the bloodstream. So, for example, oral drugs must pass through digestive system. Distribution, how the drug spreads to tissues and organs. Metabolism, how the drug is chemically modified, mainly in the liver. Excretion, how the drug is eliminated from the body through urine or bile usually. So these parameters helps to determine dosage, frequency of administration, and potential toxicity of a drug. For example, a drug may be effective in vitro, but if it is rapidly metabolized in liver, it becomes clinically useless. So a good drug is not just the one that works, it is the one that reaches the right place and stays long enough and it is safely removed. So at preclinical stages itself, many drug candidates are rejected, and it is rejected mainly due to its high toxicity, poor bioavailability, rapid degeneration, or maybe due to its off target effects. So, this stage is essentially a safety filter. Only compounds with acceptable safety and pharmacokinetic profile moves forward. It is also important to mention the clinical aspects of the preclinical study. So, animal experiments are conducted under guidelines, very strict guidelines. That is, we have to minimize the animal use and we have to reduce the suffering. So, so we follow three R's, that is replacement, reduction, and refinement. And in order to replace this animal system, modern approaches are also used, like organoids, organ on chip system, as well as computational models. So to summarize, preclinical studies, it is done to ensure a drug is biologically active, pharmacokinetically stable, and most importantly, so after passing the preclinical evaluation, a drug candidate is finally ready for the most critical stage, that is the clinical trials, where it is actually tested in human. So this stage represents the transition from laboratory science to real world medical application. At this point, the key question becomes, will this drug actually work safely in human? Clinical trials are systemic control studies conducted in human participants to evaluate safety, efficiency, dosage, and side effects. They are conducted in multiple phases, each designed to answer a very specific scientific and clinical question. Importantly, clinical trials follow strict ethical and regulatory guidelines to ensure participant safety. So, clinical trials are divided into four stages. Phase 1, which tests the safety of the drug. Phase 2, which tests the efficiency of the drug. Phase 3, which acts as the confirmation of the drug. And Phase 4, which 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 targets the post-marketing surveillance of the drug. Each phase involves a larger number of participants and provides deeper insights into the drug's performance. So let's begin with phase 1 trial. This is the first time the drug is tested in human. So it is conducted in 10 to 100 healthy volunteers, focuses on safety and tolerability. So its main objective includes determining the safe dosage range, identifying the immediate side effects, and studying the pharmacokinetics in humans. So researchers carefully monitor the heart rate, blood pressure, and organ functions of the healthy individuals participating. So at this stage, the goal is to see if the drugs work, but to ensure that it is safe enough to proceed further. Once the safety is established, the drugs moves to phase 2 trials. It is conducted in 100 to 500 patients. Participants actually have the disease, and the objective of the phase 2 involves to evaluate the efficiency, identify the short term side effects, and to optimize the dosage. So, this is the stage where the drug is tested for its therapeutic effect. For example, a drug cancer. Sorry, does a cancer drug reduce a tumor size? Does an antibiotic eliminate the infection? So, this is where the most drugs try fail. Even if a drug shows excellent results in preclinical studies, it may not produce the desired effect in human. Why this failure happened? It is because disease complexity in human, biological variability, or maybe our incomplete understanding of the mechanism. So phase 2 answers the most important question in the drug trial. That is, does the drug actually work in patients? So after demonstrating efficiency, the drug proceeds to phase 3 trial, and it is conducted in 1000 to 5000 patients, and it is multi-centered, and it is often across different countries. So the phase 3 objective include confirm the effectiveness, compare with the existing treatment, monitor the adverse effects. So this phase provides statistically significant data required for regulatory approval. So the main study design elements of phase 3 includes randomization, and it includes a control group, that is a placebo or standard treatment group, and also the blinding. It may be a single blind or a double blinding. So why it is important? Because it reduces the bias and ensures a reliable result. So phase 3 answers the question, that is this drug better or at least capable of, at least comparable to the existing treatment. So, even after the approval, monitoring does not stop. This is the phase 4 trial or the post marketing surveillance. So, its objective is to detect a rare or long term side effects, evaluate real world effectiveness, monitor the drug interaction. So, for example, side effects of some of the drugs only appear after years of use or in specific population. So phase 4 ensures that the drug remains safe even after widespread use. So one of the most striking facts in drug discovery is this: nearly 90% of drug candidates fail during the clinical trials, especially during the phase 2 trial. And it may fail due to various criteria, mainly due to its lack of efficiency, or maybe its unexpected toxicity, or poor pharmacokinetic performance, or variability in patient response. So, this highlights a fundamental challenge. Human biology is far more complex than an the regulatory approval and drug design. So, after the drug has passed through clinical trials, it has to be approved by certain agencies. It include FDA, EMA, CDSCO. CDSCO is a regulatory body ensuring a drug is safe for Indian population. So these regulatory bodies ensure and evaluate safety, efficiency, and manufacturing quality before guaranteeing a market's authorization. And there is also modern drug design approaches, which is based on structure based design, or maybe ligand based design, or AI driven discovery. So now we have discussed how drugs are developed and tested. So let's move on to a very equally important question. What type of therapies are we actually developing? This brings us to therapeutic modalities, which represent different strategies used to treat diseases. So, what are therapeutic modalities? So, these refer to different categories of treatment approaches used in modern medicine. Over time, drug discovery has evolved from simple chemical drugs to highly advanced and personalized therapies. Today, the major modality include small drug, small molecule drugs, biologists, and gene and cell therapies. So first, we are going to discuss about small molecules. So these are molecules of lower molecular weight, and it is chemically synthesized, and it is usually administered orally. So its mechanism includes inhibiting enzymes, maybe blocking receptors, or modulating signaling pathways. So its advantages are it is easy to manufacture, it is stable, cost effective, and it can enter the cells effectively. So the next one is biologists, which are more advanced therapeutics when compared to small molecules. So these are large complex molecules and it is derived from living systems rather than from a chemical derivatives. Unlike of small molecules, it includes proteins, peptides, and antibodies. So its mechanism is very specific and it often binds to a single targets and it blocks a specific signaling pathway. So the last and the most advanced is the gene and cell therapy. So the gene therapy involves modifying or replacing a defective gene and it uses tools like CRISPR-Cas. And the cell therapy, it is used in living cells as treatments. That is, we take the living cells and we genetically modify and it is administered back into the patient for the treatment and it is mainly used in cancer cells, for example CAR T cell therapy, and it has got more advantages than small molecules and biologists, that it targets the root cause and it is potential for long term or permanent cure. So these therapies are not just treating the symptoms, they are rewriting the biology itself. So, looking into the major challenges in drug discovery. So, the main challenge is the cost. So, developing one drug costs about 1 to 2 billion US dollars. So, the next challenge is the high failure rate. The most drugs fail during the clinical trials, especially during the phase 2. The third limitation faced by the scientific community is the drug resistance, especially in microbe related drug discovery and cancer related. The targets evolve in both of the diseases, and drugs become ineffective as the year progresses. So, the next is the complexity of the human, that the human body is variable from human to human, and the drugs that may be effective in the preclinical studies may not be effective in human due to its advanced molecular variability, and the diseases involve multiple pathways. And lastly is the safety concerns. A drug may cause side effects and it can cause a long-term toxicity. So these are the few challenges faced by the scientific community during a drug discovery. So to summarize, therapeutic modalities have evolved from simple chemical drugs to highly sophisticated biologies and genetic intervention. At the same time, challenges like cost, resistance, and complexity continues to shape the future of drug discovery. The first one is AI and machine learning. So AI can analyze massive data sets such as much faster than traditional methods, making the discovery process more efficient. Personalized medicine. Instead of a one size fits all approach, therapies are tailored to improve the effectiveness and reduce the side effects. This is especially important in cancer treatments. The third one is nanotechnology. This enables a targeted drug delivery using nanoparticles. So, this allows drugs to reach a specific tissue, improving efficiency and minimizing off target toxicity. It is widely being explored in cancer therapy and vaccine delivery. Finally, we have immunotherapy, and it is mainly used in cancer treatment, and examples include CAR T, cell therapy, and immune checkpoints, and these approaches have shown remarkable success, particularly in cancer treatment. So overall, these advancements are making drug discovery faster, more precise, and more patient specific, representing a major shift towards next generation therapeutics. Now, to conclude, drug discovery is a complex, multi step process that transforms a simple biological idea into life saving therapy. It begins with understanding the disease at molecular level, followed by identifying the right target, designing, optimizing drug candidates, rigorously testing them through preclinical and clinical stages. Despite the long timeline, high cost, and significant failure rates, this process ensures that only the safest and most effective treatment reach the patient. What is particularly exciting is that the field is continuously evolving. Advances in AI, personalized medicine, nanotechnology, and immunotherapy are making drug discovery faster, more precise, and more efficient than ever before. So, ultimately, drug discovery is not just about developing new drugs, it is about improving quality of life, advancing healthcare, and addressing unmet medical needs. With that, I would like to thank you all for your attention. Thank you.
Thank you, Ashley, for that clear and informational lecture. As we continue our discussion on therapeutic development and modern drug discovery, it is also important to understand how different human diseases often share interconnected molecular pathways. Our next lecture will be delivered again by Aditi Dubey. Her lecture is on From Cancer to Alzheimer's, Common Molecular Mechanisms in Human Disease. Welcome, Aditi.
Good afternoon, everyone. I'm Aditi Dubey, and today it's an honor to present a topic that brings together oncology, neuroscience, and modern molecular biology. My presentation is titled From Cancers to Alzheimer's, Common Molecular Mechanisms in Human Disease. At first, these two diseases seem completely unrelated. Cancer is associated with uncontrolled growth and excessive survival of cells. Alzheimer's disease is associated with neuronal dysfunction, memory decline, and progressive cell loss. So, one disease appears to involve too much growth while the other involves too much degeneration. However, modern research shows that beneath these opposite clinical outcomes lie many shared molecular disturbances. Processes such as oxidative stress, inflammation, mitochondrial dysfunction, altered metabolism, and DNA damage are central to both diseases. Today, I will show how understanding these common mechanisms helps us develop a more integrated view of disease biology. And to begin that journey, let us first ask an important question. Why should we even compare cancer and Alzheimer's? This comparison is valuable because medicine has traditionally studied diseases separately. Cancer is usually placed in oncology, Alzheimer's is usually placed in neurology, but cells in every organ rely on the same core biological systems. They all need energy, they all repair DNA, they all regulate proteins, they all respond to stress, they all communicate with the immune system. When these systems fail, disease emerges. The only difference is how that failure is expressed in different tissues. For example, in a dividing tissue, loss of control may lead to uncontrolled proliferation. In the brain, loss of control may lead to neuronal dysfunction and degeneration. So instead of seeing these diseases as unrelated, we can see them as different manifestations of disrupted cellular homeostasis. This leads us to an even broader concept. Human disease is often the result of network failure. Modern biology increasingly recognizes that diseases are not caused by one isolated event. They usually arise when interconnected biological networks become unstable. These networks include DNA repair pathways, mitochondrial energy systems, protein quality control pathways, stress signaling pathways, immune regulation, and metabolic circuits. When one pathway is disturbed, others are affected as well. That is why chronic diseases are often complex and progressive. For example, mitochondrial dysfunction can increase oxidative stress. Oxidative stress can damage DNA. DNA damage can trigger inflammation, and inflammation can further damage tissues. So, disease is often a cascade rather than a single step. With that systems view in mind, let us now look at the first shared mechanism, that is, genomic instability. Every day, our DNA faces constant stress from replication errors, environmental toxins, radiations, and normal metabolism. Fortunately, cells possesses powerful DNA repair systems. But when these repair systems weaken, genomic instability develops. In cancer, genomic instability is a major engine of disease progression. Mutations accumulate, growth pathways activate, and tumor suppressor system fail. In Alzheimer's disease, neurons also accumulate DNA damage with age. Although neurons do not divide frequently, they are long lived and metabolically active, making genomic integrity essential. Over time, unrepaired DNA damage may impair neuronal function and survival. So, in both diseases, damaged DNA is important. But because the cell types are different, the outcomes are different. Now, one major cause of DNA damage is oxidative stress, which brings us to our next mechanism. Oxidative stress occurs when reactive oxygen species exceed the cell's antioxidant defenses. Reactive oxygen species, or ROS, are naturally produced during metabolism. At controlled levels, they are useful signaling molecules. But when excessive, they damage DNA, proteins, lipids, membranes, and mitochondria. In cancer, oxidative stress may promote mutations and tumor evolution. Cancer cells often adapt by increasing antioxidant systems. In Alzheimer's disease, oxidative stress injuries neurons and synapses, contributing to cognitive decline. So, although the outcomes differ, oxidative stress is a shared biological pressure. And importantly, one of the main sources and targets of oxidative stress is the mitochondrion. Mitochondria are best known for ATP production, but they also regulate many essential processes. These include energy metabolism, calcium signaling, ROS generation, apoptosis, cellular adaptation. In cancer, mitochondria are often reprogrammed to support rapid growth. They help generate building blocks needed for dividing cells. In Alzheimer's disease, mitochondrial decline reduces neuronal energy supply. Since neurons require large amounts of energy, even modest dysfunction can have major consequences. Reduced ATP, increased ROS, and impaired signaling can all damage brain function. So, mitochondria can either be hijacked for excessive growth or fail during degeneration. Another process strongly linked to mitochondrial dysfunction is chronic inflammation. Inflammation is beneficial when temporary. It helps fight infection and repair injury. But when inflammation becomes chronic, it can drive disease. In cancer, inflammatory molecules can stimulate cell proliferation, angiogenesis, tissue invasion, and immune evasion. In Alzheimer's disease, chronic activation of brain immune cells called microglia contributes to neuronal stress and synaptic damage. Thus, in both diseases, inflammation changes from a protective process into a harmful one. And persistent inflammation also disrupts protein quality control, which brings us to the next mechanism. Cells must continuously maintain protein quality. Proteins need to be correctly synthesized, folded, repaired, and degraded. This balance is also known as proteostasis. When proteostasis fails, toxic proteins accumulate. In Alzheimer's disease, this is highly visible through amyloid beta plaques and tau tangles. These abnormal proteins interfere with neuronal communication and survival. In cancer, proteostasis is also crucial. Cancer cells experience high metabolic stress and produce many abnormal proteins. To survive, they rely heavily on chaperones and degradation systems. So, while protein aggregation is more famous in neurodegeneration, protein quality control is equally important in cancer biology. This naturally raises a central question. Why do some cells survive too much while others die too easily? Healthy tissues depend on balanced cell fate decisions. Cells must know when to divide, pause, repair damage, or undergo programmed death. In cancer, these control systems fail in one direction. Cells evade apoptosis and continue proliferating despite damage. In Alzheimer's disease, failure occurs in the opposite direction. Neurons progressively lose function and die. So we can think of cancer as excessive survival and Alzheimer's as insufficient survivals. Both are failures of regulation. This balance is also strongly influenced by aging, which is one of the biggest risk factors for both the diseases. Age is the strongest common risk factor for both cancer and Alzheimer's disease. As we age, cells accumulate DNA damage, oxidative stress, mitochondrial decline, senescent cells, chronic inflammation, and reduced repair capacity. These changes create biological vulnerability. In tissues that continue dividing, this may favor cancer. In highly specialized long lived cells such as neurons, it may favor degeneration. So, aging is not a single disease. It is a background state that increases risk across many diseases. Another important age related factor is metabolic change. Metabolism determines how cells generate and use energy. In cancer, cells often shift toward high glucose consumption and biosynthesis. This supports growth and rapid division. In Alzheimer's disease, the opposite problem is common. Affected brain regions often show reduced glucose utilization. This energy shortage can impair neuronal signaling and resilience. So, both diseases involve metabolic disruption. Cancer shows excessive growth metabolism, whereas Alzheimer's shows failing neuronal metabolism. Metabolism and immunity are also closely connected, which leads us to the immune system involvement. The immune system plays a major role in both diseases. In cancer, tumors often avoid immune destruction by suppressing T cell activity or exploiting checkpoints pathways. This led to immunotherapy breakthroughs. In Alzheimer's disease, dysregulated innate immunity and chronic microglial activation contribute to disease progression. So, whether the problem is immune escape or immune overactivation, the immune system is deeply involved. Now, a natural question follows. If so many pathways overlap, why do the diseases still look so different? The answer lies in biological context. The same molecular disturbance can produce different outcomes depending on cell type, tissue environment, regenerative capacity, metabolic demand, and age of the tissue. Let's see an example. A mutation in a rapidly dividing epithelial cell may promote tumor growth. The same stress in a neuron may impair function and trigger degeneration. Therefore, pathways may be shared, but context determines disease expression. Recognizing this helps researchers learn across specialities. Cross disease thinking has great value. Cancer research has advanced precision biomarkers, genomic testing, AI diagnostics, and adaptive therapies. These tools may help neurology. Meanwhile, neurodegeneration research offers insights into protein aggregation, cellular resilience, aging biology, and long term disease progression. Instead of separate silos, medicine benefits when fields learn from each other. This is the heart of systems biology. Systems biology asks us to study across multiple layers: genes, RNA, proteins, metabolites, cells, tissues, environment, and time. Rather than asking which one mutation caused disease, we ask how networks became unstable. This integrated view is especially important for complex chronic diseases, and it also shapes the future of medicine. Future healthcare may increasingly target shared mechanisms rather than isolated diagnosis. Examples include anti inflammatory prevention strategies, mitochondrial support therapies, senescence targeting drugs, AI based risk prediction, early biomarker screening, and personalized lifestyle interventions. This means one intervention may reduce risk across multiple diseases. That is the promise of integrated disease biology. Now, let me summarize the key messages. Cancer and Alzheimer's appear opposite clinically but share important molecular pathways, which include oxidative stress, inflammation, DNA damage, mitochondrial dysfunction, metabolic disruption, and aging biology. The final disease depends on tissue context. Understanding these shared mechanisms can accelerate better prevention and treatment. Now, to conclude, I would just say to truly understand disease, we must look beyond organs and beyond labels. We must recognize that cancer, Alzheimer's, diabetes, cardiovascular disease, and many others are connected through common biological principles. When we understand those shared principles, medicine becomes more predictive, more integrated, and more powerful. That may be the future of biomedical sciences. Thank you very much for your attention throughout the lecture.
Thank you, Aditi. That was a very insightful lecture. We now move toward computational and bioinformatics driven approaches in therapeutic development. Our next lecture will be delivered by Kompala Jhansi, who is currently pursuing a Bachelor's of Engineering in Bioinformatics. Her lecture is titled Bioinformatics Driven Drug Discovery From Target Identification to Lead Discovery. Welcome, Jhansi.
Hello everyone. Firstly, thank you all for being here today. My name is Jansi, and it gives me a great pleasure to present my work on one of the most dynamic and promising intersections of modern science, which is bioinformatics driven drug discovery from target identification to lead discovery. In the next 20 minutes, we will embark on a comprehensive and detailed journey through the modern drug discovery pipeline. We will examine the persistent challenges that had plagued traditional pharmaceutical development for decades, explore how bioinformatics and computational biology are revolutionizing every stage of the process, analyze powerful real world success stories, and finally look at the promising horizon shaped by artificial intelligence and machine learning. Drug discovery sits at the intersection of biology, chemistry, data science, and medicine. It represents humanity's most sophisticated attempt to translate fundamental biological understanding into tangible therapeutic solutions that alleviate the suffering and save lives. However, as we all know, this translation has historically been fought with enormous technical, financial, and temporal difficulties. By the end of this presentation, I hope you will have gained not only a clear conceptual understanding of drug discovery pipeline, but also a deep appreciation for the critical role that bioinformatics plays in making the entire process more rational, efficient, predictive, and ultimately successful. I encourage you to ask questions at the end, as I believe the most valuable insight often emerge through active discussion. With that, let us begin by confronting the sobering realities of traditional drug discovery. Let us first acknowledge the scale of challenge before us. Drug discovery is widely regarded as one of the most difficult and resource intensive scientific undertakings in human history. According to industry experts, the average time required to bring a new drug from initial discovery to final regulatory approval ranges between 10 to 15 years. The total capitalized cost frequency exceeds $2 billion per approved therapeutic agent. Even more concerning is the staggering 90% clinical failure rate that is observed across the industry. This means that out of every 10 drugs that enter the human clinical trials, only one ultimately reaches the market. These failures are not just random. Many candidates fail due to insufficient efficacy in humans despite promising the preclinical data. Others are halted because of unexpected toxicity, poor pharmacokinetic profiles, inadequate bioavailability, or failure to outperform the existing standard of care treatments. The economic and human cost of these failures is immense. Late-stage clinical trial failures, in particular, represents the massive financial losses and delayed hope for patients who are suffering from serious diseases such as cancer, neurodegenerative disorders, rare genetic conditions, and infectious diseases. Historically, drug discovery relied heavily on phenotypic screening and natural product extraction. While these empirical approaches yielded many important medicines, including penicillin, aspirin, and numerous chemotherapeutics, it is inherently inefficient in today's data rich environment. The pharmaceutical industry now generates enormous volumes of biological data every day through natural product screening, new generation sequencing, high throughput proteomics, single cell transcriptomics, CRISPR based functional genomics, and even advanced imaging techniques. Without these sophisticated computational frameworks to organize, integrate, analyze, and interpret this data, researchers would be overwhelmed by its sheer volume and complexity. This is where the bioinformatics emerges as a game changing discipline. Bioinformatics proves the computational infrastructure and analytical methodologies that are necessary to convert the raw biological data into the actionable biological insights. It enables the researchers to identify causal disease mechanisms, prioritize the most promising therapeutic targets, predict drug target interactions, optimize the molecular structure, and reduce attrition rates at every stage of development. In essence, bioinformatics is shifting the paradigm from a trial and error model to a predict and validate model. This transition is not merely incremental, it represents a fundamental transformation in how we approach the therapeutic innovations in the 21st century. The integration of multiomics data, structural biology, machine learning, and artificial intelligence is helping pharmaceutical companies and academic researchers make smarter decisions early in the pipeline, thereby saving their time, reducing cost, and most importantly, accelerating the delivery of life changing medicines to patients who urgently need them. With this context established, let us now examine the structured stages of the modern drug discovery pipeline and understand precisely where and how bioinformatics exerts its greatest influence. The contemporary drug discovery and development pipeline is typically conceptualized as a seven-stage process. While these stages are presented sequentially, the reality is often iterative, with feedback loops connecting the different phases. The pipeline begins with disease biology understanding and target identification, followed by target validation, hit discovery, lead optimization, preclinical testing, clinical trials from phase 1 to 3, and finally regulatory approval and post market surveillance. In today's presentation, I will primarily focus on the early discovery phases from target identification to lead discovery because this is where bioinformatics and computational approaches deliver the highest return on investment by dramatically improving decision quality and reducing downstream failure rates. Each stage builds upon the previous one, with rigorous go or no go decision points designed to eliminate the weak candidates as early as possible. The integration of computational tools throughout this pipeline has significantly enhanced the predictability and efficiency. Let us now explore these stages in detail, beginning with the foundational step, target identification. Target identification represents the critical first decision point in the entire drug discovery process. The fundamental scientific question is that which scientific biological molecules, such as genes, proteins, or pathways, that are casually driving the disease process? In the pre-genomic era, target identification relied largely on limited experimental observations and educated guesses. Today, thanks to bioinformatics, we have access to the unprecedented wealth of multi-layered biological data that can be systemically integrated and analyzed. The input data layers are as follows. Firstly, genomics. Genomics provides insights into disease associated genetic variants, single nucleotide polymorphisms, and genome wide association study signals. Secondly, transcriptomics. Particularly, RNA sequencing and single cell transcriptomics reveals differential gene expression patterns between healthy and diseased states, highlighting that genes are upregulated or downregulated in pathology. Next, proteomics. Proteomics offers information about actual protein levels and post translational modifications, followed by protein protein interactions and subcellular localization. At last, pathway analysis. Pathway analysis and network biology tools helps us understand how these individual components interact within complex cellular signaling cascades and metabolic networks. Modern drug discovery bioinformatics platforms integrate these diverse data using sophisticated algorithms, including graph theory, machine learning based ranking model, and Bayesian integration frameworks. Targets are prioritized based on multiple weighted criteria. First, strength of genetic association. Next, disease specific expression patterns. Next, druggability potential, which shows the presence of suitable binding pockets. Next, network centrality. Next, essentially scores and predict safety profiles. The output of this stage is carefully curated. Ranked list of high confidence candidate targets are then taken, and these are no longer vague hypotheses but biologically well supported molecular entities with strong mechanical rationale. This system level approach significantly improves the quality of targets entering the pipeline and helps avoiding pursuing molecules that, while biologically interesting but may not be therapeutically tractable. Next, move on to target validation. Target identification must be followed by rigorous target validation. Here comes the interesting question. Does the target truly matter? And then computational target validation has become exceptionally powerful. Key tools and databases includes string and other interaction databases that construct the comprehensive protein-protein interaction networks, revealing the functional relationships and potential compensatory pathways. Next, AlphaFold. AlphaFold is a revolutionary deep learning system that predicts protein three-dimensional structure with atomic level accuracy, even for the proteins that are never before experimentally categorized. Next comes molecular docking. Molecular docking simulates ligand binding poses and estimates the binding affinity at the target site. At last, binding pocket detection. Binding pocket detection algorithms includes fpocket, sitemap, and cavity that identify and characterize druggable cavities based on geometric and physiochemical properties. These tools allow researchers to perform in silico validation before committing expensive experimental resources. Targets that demonstrate strong disease linkage, favorable druggability characteristics, and low predicted off target risks are advanced with much higher confidence. This stage has dramatically reduced the number of targets that fail in later, more expensive phases of development. Next, step 3, hit and lead discovery. Once a target is validated, the search begins for chemical compounds, which are hits, that can bind to and modulate the target effectively. Here comes the virtual screening. Virtual screening has revolutionized this stage. Researchers can now computationally screen millions to billions of compounds for commercial and proprietary chemical libraries in a matter of days or even weeks, compared to months or years required for traditional high throughput screening. Two major approaches are used here. First is structure-based virtual screening. Second is ligand-based virtual screening. In structure-based virtual screening, docking large libraries into the 3D structure of the target is done. And in ligand-based virtual screening, they use known active molecules as templates for the similarity screening. And next, each ADMET property, absorption, distribution, metabolism, excretion, and toxicity prediction models help eliminate the compounds with problematic pharmacokinetics or safety profiles. The goal is to identify a small number, typically 3 to 5, of high quality hit compounds that show promising activity and drug like properties for the future optimization. Next comes step 4, lead optimization. Hit compounds are starting points, are not the finished drugs or anything. The lead optimization phase involves iterative chemical modifications to transform the promising hits into robust clinical candidates. Scientists focus on optimizing the four key parameters: potency, selectivity, stability, and safety. First, potency. By improving the binding affinity and functional activity, potency is achieved. And next, selectivity. It is achieved by reducing unwanted interactions with off-target proteins. And third, stability, which is by enhancing the metabolic stability and pharmacokinetic properties. And at last, safety. It is by minimizing the toxicity risks and reactive metabolite formation. This entire process is guided by structure activity relationship studies and supported by computational modeling tools. Each cycle of design, synthesis, testing, and analysis brings the molecule closer to the ideal profile required for the clinical advancement. The integration of these computational predictions with the experimental data creates a powerful feedback loop that accelerates this traditional time consuming phase. Next, move on to step 5, preclinical and clinical translation. Even the more sophisticated computational predictions must ultimately be validated in biological systems. The translational phase bridges the gap between in silico work and real world applications. It progresses systematically through in vivo cell based assays, in vivo animal models, and then carefully structured phase 1, 2, and 3 clinical trials. Bioinformatics continues to provide critical support during these stages through biomarker discovery, patient stratification using genome profiles, pharmacogenomics, and post-approval pharmacovigilance. This seamless integration of computational and experimental approaches represents the true power of modern translational medicine. Now, let's see the case study of imatinib that is marketed as Gleevec. One of the most compelling demonstrations of bioinformatics enabled drug discovery is the development of imatinib. Imatinib was rationally designed to target the BCR-ABL fusion kinase created by the Philadelphia chromosome that is translocated in chronic myeloid leukemia. Using crystallographic data, computational modeling, and structure guided designs, researchers developed a highly selective inhibitor that binds to the ATP binding pocket of the aberrant kinase. This precision targeting transformed CML from a fatal disease with poor prognosis into a manageable chronic conditions. The 10-year survival rate increased dramatically from approximately 20% to 80%. Imatinib remains a landmark achievement in precision oncology and a powerful validation of the target based, structure guided approach to the drug discovery. And one of the most exciting parts of this presentation is the transformative role of artificial intelligence in drug discovery. AI is not merely an incremental improvement or a supporting tool. It represents a fundamental paradigm shift, a complete rewriting of how we discover and develop the new medicine. What took traditional method decades can now, in many cases, be accomplished in months or even weeks with AI powered approaches. Let us elaborate on major applications of AI across the drug discovery pipeline. First, target prioritization and identification. One of the earliest and most impactful applications of AI is in intelligent target prioritization. Traditional target identification relied on manual curation of literature and limited experimental data. Today, machine learning models can integrate hundreds and thousands of data points from various sources like genomics, transcriptomics, proteomics, etc. simultaneously. And advanced deep learning architectures, including graph neural networks and transformer models, can now analyze complex biological networks to identify the most promising therapeutic targets. And these models consider not just statistical association but also causal interference. For example, AI system can now predict which protein are most likely to be druggable and causally linked to the disease while minimizing the risk of side effects. This capability is particularly valuable for complex diseases like Alzheimer's, certain cancers, and rare genetic disorders where traditional approaches have struggled for decades. And secondly, structure prediction, which is also the AlphaFold revolution. Perhaps the most famous AI breakthrough in biology is AlphaFold 2 and now AlphaFold 3, which are developed by DeepMind. Before AlphaFold, determining the 3D structure of a protein through experimental methods like X-ray crystallography or cryo-electron microscope could take months to years and cost hundreds of thousands of dollars. And AlphaFold can now predict highly curable protein structures in a matter of hours, often with an atomic level precision. This breakthrough has enormous implications for drug discovery. With accurate 3D structure available for nearly all the human proteins, structure based drug design becomes possible even for previously uncategorized or undruggable targets. Let's move on to the generative chemistry, which includes admit, toxicity, and safety prediction. Predicting how a molecule will behave in a human body is one of the biggest bottlenecks in the drug development. AI models trained on massive data sets of experimental results can now predict admit properties, HERG liability, hepatotoxicity, cardiotoxicity, and other safety concerns with impressive accuracy. Early elimination of problematic molecules saves enormous resources and reduces the risk of late stage failures. And at last, patient stratification, which includes the clinical trial optimization as well. AI's impact extends well beyond early discovery. In clinical development, machine learning is used for patient stratification, identifying the subgroups most likely to respond to a therapy based on its genomic, proteomic, and clinical biomarker. Next, adaptive trial design, dynamically adjusting the trial parameters based on the incoming data. And at last, real world evidence analysis and post marketing surveillance. This precision approach significantly improves the clinical success rates and supports the vision of personalized medicine. We begin with biological sequence data such as DNA and protein sequences to identify the gene or protein that are involved in disease. Once a promising target is identified, computational tool help predict its three-dimensional structure, allowing the researchers to locate the binding sites where drug molecules can interact. Next comes function. Understanding how the target contributes to disease pathways and whether modifying it can produce a therapeutic effect. Finally, we move forward towards medicine, where candidate molecules are virtually screened, optimized, and prioritized before experimental validation. And there are four important key takeaways. First, computational biology reduces cost and uncertainty by improving early decision making. Second, bioinformatics helps prioritize the most relevant and druggable targets. Third, virtual screening accelerates the hit discovery by narrowing large chemical libraries into promising candidates. And finally, artificial intelligence is reshaping the modern therapeutics by making drug discovery faster, smarter, and more precise. In summary, bioinformatics has transformed the drug discovery from a high risk empirical process into a data driven, computationally guided discipline. The integration of genomics, structural biology, virtual screening, and AI is accelerating the path from biological insight to patient impact. In short, today's medicine increasingly begin as biological data and evolve through computational intelligence into therapeutic solutions. And thank you so much for this opportunity to present on a drug discovery pipeline.
Thank you, Jhansi, for that comprehensive lecture. Continuing our discussion on therapeutic strategies, we once again welcome Shreya Das. Her lecture is on From Vulnerability to Drug, Exploiting Cellular Stress Dependencies in Therapeutic Design. Welcome, Shreya.
Good afternoon, everyone. This is my second lecture for today, and here I want to take us from a biological observation to a therapeutic strategy. And the observation is simple. The same stress pathways that help disease cells survive can also become their most actionable liabilities. This lecture is called From Vulnerability to Drug, Exploiting Cellular Stress Dependencies in Therapeutic Design. And the central question here is not whether stress responses matter, because by this time we know that they do. The real question comes up like how we convert this biology into something druggable, selective, and clinically meaningful. Across the next several slides, I will argue that stress biology becomes medicine only when vulnerability is translated through structure, selectivity, modality choice, and patient context. So, before we talk about drugs, I want to make sure the foundational logic is completely solid, because everything that follows in this lecture completely depends on it. So, here is the core idea. Stress response pathways are conserved. That means evolution has preserved them across billions of years, across organisms, across cell types. These are not optional programs, rather, they are mandatory survival infrastructure. When a cell faces proteotoxic load, oxidative damage, nutrient deprivation, or even replication pressure, it activates these circuits because it has no other choice. They are the default response to threat. Now, and this is the critical point, in a healthy cell in a normal physiological context, these pathways are activated briefly. They do their job and again return to baseline. They are acute, reversible, and even tightly regulated. So we can say that the cell uses them the way you use an emergency generator. You turn it on during a power cut, it keeps the lights on, and then you turn it off when the main power gets restored. But we should think about like what happens in disease. In disease, the analogy of power cut says like the power cut can never end. In cancer, the cells face chronic proteotoxic stress from oncogene-driven proliferation, hypoxia, nutrient scarcity, and genomic instability. They live with permanently activated stress responses. And because natural selection operates on tumor cell populations, the cells that survive, they are actually the ones that have hardwired the stress responses, that have made the emergency generator a permanent part of the electrical grid. In neurodegeneration, something similar but opposite happens. Neurons accumulate the misfolded proteins over decades. They face chronic oxidative stress from the extraordinary metabolic activity. They activate the same stress circuits, but they are post-mitotic. They cannot divide and dilute the damage. In fact, they cannot adapt forever as well. And eventually, the circuit breaks. In both the cases, cancer and neurodegeneration, in fact, in infectious diseases as well, the stress response pathway is the center of the story. Luo and colleagues in Cell 2009 had articulated this as the concept of non-oncogene addiction. That is, the idea that disease cells become exquisitely dependent on stress support pathways that normal cells can tolerate losing. However, Walter and Ron had mapped the UPR as a central example of this principle. What this means for us today is very simple. The shared stress logic across diseases is not just biologically interesting. In fact, it is therapeutically actionable as well, because a dependency is a vulnerability, and a vulnerability, if it meets the right criteria, can become a target. The next slide says like why survival pathways become vulnerabilities. At the first glance, it may seem very paradoxical to you. But what you are looking at is a core conceptual inversion that makes stress-targeted drug discovery possible. Normal cells activate the stress pathways in response to acute stress. Then, once the threat gets resolved, the pathway downregulates. The cell returns to baseline and the pathways again is indispensable. It is not essential, rather it is just available. Disease cells, however, are very different. Since they live under chronic stress, because their buffering systems are continuously engaged, they have become dependent on those pathways, not as emergency responses, but in fact as core operating machinery. The pathway that was once optional has now become load bearing. It is therefore an essential infrastructure. Here comes the therapeutic opportunity. Suppose, if you remove a pathway that is essential to the disease cells but dispensable to the normal cells, you can achieve selective killing. It is not because of the chemistry, we should always remember that. Not because of a clever molecule even, but it is because of the underlying biological asymmetry. So this is actually a very profound insight. Selectivity in drug design is usually thought of as a chemical problem. Can you make a molecule that binds your target and not the others? And that is partly true. We can always agree to that. But the deeper selectivity, that is the biological selectivity we are talking about here, has to come from the disease state itself. A tumor cell living at the edge of its adaptive threshold collapses when the UPR support pathway is removed. However, a normal cell does not care if the UPR support pathway gets inhibited because it hardly needs it. So the survival pathway becomes a vulnerability precisely because it has been pressed into continuous service. Now, let me introduce you a vocabulary distinction that will be very critical for the rest of this lecture. And frankly, it is a distinction that separates rigorous target identification from wishful thinking. So the difference comes between activation and dependency. For example, when you sequence a tumor, you will find dozens and even sometimes hundreds of pathways that are differentially activated compared to normal tissue. Stress response pathways, growth factor signaling, inflammatory programs, metabolic programming, all of them are on. All of them might look like targets on a pathway map, but activation is not the same as dependence. A pathway can be active in a disease context as a bystander, elevated, measurable, but not essential. If you inhibit that, nothing dramatic will happen. The cell buffers the loss through parallel routes and carries on. This is the graveyard of drug discovery, targeting pathways that are activated in disease but not selectively required. Dependency is a totally different concept. A dependency means that the disease cell and not the normal cell requires that pathway to stay alive. That means if you remove it, the disease cell collapses, while the normal cell adapts or even simply does not notice. Chronic stress is the mechanism that actually creates these kind of dependencies. When a cell lives under sustained proteotoxic load, it rewires its internal circuitry towards the pathways that support survival. Those pathways become hypertrophied, overrepresented in the signaling network, more deeply connected, in fact, more difficult to replace. And as a consequence, the cell loses the buffering flexibility. It cannot reroute around those pathways as easily a normal cell can. It has traded versatility for specialization, and that specialization has now become its weakness. So the first question you must ask before committing to any stress-targeted program is not, is this pathway active? The question is this pathway something the disease cell cannot function without? Dependency and not activation is the key signal for intervention. Let me now make the therapeutic window concept concrete, because this is the argument you will need to make every time you propose a stress-targeted therapy, whether in a grant or a paper or in fact in a conversation with a clinician. The question will always arise like this. Why will your drug kill the disease cell only and spare the normal cell? The traditional answer is chemical selectivity. Your molecule binds target A and not target B. And target A is only expressed in tumor cells, but for stress-targeted approaches, the answer is partly biological. Suppose, think of it in this way. Normal cells have a stress baseline. They are not stressed in the pathological sense. They handle routine proteotoxic and oxidative load, buffer it efficiently, and in fact operate with plenty of margin before they reach a dangerous threshold. Howsoever, the disease cells, be it your tumor cells, your degenerating neurons, your chronically infected macrophages, they already operate close to that threshold. They're buffering very hard just to stay alive at that baseline. So they have got very little margin left. Now, when you introduce a drug, the drug increases the stress load, or in fact, it removes the buffering pathway. In a normal cell, this perturbation is absorbed. The cell activates a compensatory response, restores the balance, and hence remains fine. In the diseased cells, which was already near its threshold, the same perturbation is not absorbed. Rather, it tips the cell over the edge. It leads to threshold collapse and, as a result, death. This is why exactly the therapeutic window in stress-targeted therapies is partly a biological property of the disease state itself. The selectivity is co-created by the pathology itself. The drug doesn't need to be exquisitely selective. The disease cell's pre-existing fragility does part of the work. This is also why biomarker-selected patient populations respond better to stress-targeted approaches. If you can identify patients whose tumors are already near the adaptive threshold through high baseline ER stress signatures, elevated NRF2 activity, or in fact markers of metabolic stress, those are the patients for whom a small perturbation will be lethal. The selectivity argument depends on being able to identify where on the stress curve the patient's cell sit. In the previous lecture in the afternoon, we have already examined each of these axes in mechanistic depth. Here, I want to reframe them not as a biological phenomena, but as opportunity spaces. Proteotoxic stress. What does that mean? The accumulation of misfolded or damaged or in fact aggregated proteins that creates dependency on the UPR, on chaperone networks, on the ubiquitin proteasome system, and on autophagy. Any of these support nodes is a potential target. Oxidative stress happens when there is a chronic elevation of reactive oxygen species above the buffering capacity of normal antioxidant systems. As a result, it creates dependency on the NRF2-driven antioxidant programs, on glutathione synthesis, and on ferroptosis defense enzymes like GPX4. So these are actually the dependency nodes. Replication stress occurs when the DNA damage load comes with rapid error-prone cell division. It drives dependency on the checkpoint kinases, on DNA repair pathways, on the ATR checkpoint 1 axis. This is particularly relevant in rapidly dividing cancer cell populations. Metabolic stress, nutrient deprivation, hypoxia, altered bioenergetics, they all drive dependency on mTOR, on HIF1 alpha, on alternate metabolic routes like glutamine metabolism and 1-carbon cycling. Now, and this is a point of clinical importance, we have to always remember that these axes do not operate in isolation. They converge, amplify one another, and share regulatory nodes. A cell under oxidative stress is also under proteotoxic stress because ROS oxidizes proteins and triggers misfolding. A cell under metabolic stress is also under replication stress because energy deprivation impairs the DNA repair fidelity. This convergence is both a biological reality and in fact a therapeutic opportunity. Hitting the convergence nodes, that is the targets that sit at the intersection of multiple stress axes, tends to produce more potent and more durable responses than targeting single axis dependencies. So we have already established that dependency is what matters and not just activation. Now the question becomes very practical. Given a list of stress-dependent pathways in a disease context, how do you decide which ones are worth pursuing? Here, I can give you 3 filters. The first is selective essentiality. Is the target disproportionately essential in disease cells compared to the matched normal tissue? This is what functional genomics screen. For example, the CRISPR dropout screens, RNA interference, DepMap cancer dependency data, they are all designed to reveal a gene that is essential in tumor cells but not in normal epithelial cells is selectively essential. That asymmetry is the minimum requirement for a viable target. The second filter is the concept of non-oncogene addiction. Cancer cells are addicted not only to the oncogenes that drove the transformation, but to the stress support genes that allows them to tolerate the damage caused by those very oncogenes. A tumor driven by MYC overexpression faces massive proteotoxic stress from hyperactivated transcription and translation. That tumor is addicted to HSP90, to proteasome function, to autophagy, not because these are oncogenes, but because the oncogenic program could not operate without them. These non-oncogene additions are often more tractable than the oncogenes themselves because they are actually the enzymes with defined pockets rather than transcription factors or scaffolds. The third filter I would like to mention is the synthetic lethality. Two genes are synthetically lethal if loss of either alone is tolerable, but loss of both becomes lethal. The classic example in this case is the BRCA1-2 mutation and the PARP inhibition. In a stress biology context, synthetic lethality often involves combining a baseline elevated stress state, which you cannot alter because it is driven by the oncogenic program, with inhibition of a compensatory pathway that buffers it. This kind of combination becomes lethal. Neither alone is sufficient. So, these three filters, selective essentiality, non-oncogene addiction, synthetic lethality, they're how you convert a list of active stress pathways into a ranked list of actionable targets. A vulnerability must be reachable. Here is where many academic drug discovery programs actually fail. So, you identify a beautiful dependency. You have genetic validation. Knockdown or knockout of the target kills the disease cells selectively. You have got clinical motivation, and then you discover that your target has got no druggable binding site and the entire program stalls. So from this, we can conclude that biological importance is not enough. A target must also be chemically reachable. What makes a target reachable? The primary determinant is the presence of a druggable binding pocket, a concave surface on the protein that has the right geometry, the right electrostatic character, and sufficient depth to accommodate a small molecule and hold it with meaningful affinity. Enzymes are often naturally druggable because the active sites are precisely this: deep, defined pockets with catalytic residues whose geometry makes them ideal for small molecule engagement. Kinases, proteases, phosphatases, these are the workhorses of pharmacology because evolution has already given them these pockets. But for transcription factors, scaffolding proteins, and protein-protein interaction interfaces, it is very hard, as they tend to be flat, shallow, and conformationally dynamic. Historically, these were considered undruggable, but this is changing. Staple peptides, macrocycles, fragment-based design, and especially targeted protein degradation via PROTACs and molecular glues are opening these kind of targets. But they obviously require substantially more investment and sophistication. Context of stress biology, the most tractable targets right now include HSP90 and its core-chaperone system, the classical enzymatic architecture, well-defined ATP binding pocket, the 20S proteasome having enzymatic active sites, PERK kinase, IRE1 alpha kinase, RNase dual domain, and GPX4, which has a selenocysteine active site that can be engaged covalently. NRF2 and ATF4 as transcription factors are harder, but the upstream regulators KEAP1 and EIF2B are more tractable. One another point, conformational state matters. A protein that exists in multiple conformational states may be druggable in one state and not the other. AlphaFold2 and its successors have dramatically accelerated our ability to predict these states, to identify cryptic pockets that are only exposed transiently and to prioritize targets for structural characterization. AlphaFold and the subsequent AlphaFold 3 work have genuinely changed the early stage target assessment landscape. So, if you're designing a stress targeted program in 2026 without integrating the structural prediction, you're obviously leaving information on the table. Once you have got a tractable target, you have to choose how to engage it. And this choice, that is the drug modality, should follow the biology of the target and not the fashion of the platform. This sounds obvious, but in practice, the field does often the reverse. Suppose a laboratory with a PROTACs platform will try to degrade everything. Similarly, a laboratory specializing in covalent chemistry will find covalent targets. The modality is chosen first and the biology is fitted to it afterwards. So this is backwards. So let me give you the right framework. Orthostatic inhibition, that is, blocking the site of an enzyme, is the default approach for enzymatic targets with well-defined focus. It is mechanistically clean, pharmacologically interpretable, and structurally straightforward. PARC inhibitors, proteasome inhibitors, and in fact HSP90 inhibitors work this way. Allosteric modulation means like engaging a site physically separate from the active site and inducing a conformational change. This is valuable when the active site is occupied or you want to modulate rather than abolish the entire activity. IRE1 alpha, for example, can be targeted allosterically at its kinase domain to modulate its RNase output without fully inhibiting it, which matters because complete IRE1 alpha ablation can paradoxically reduce the ER stress resolution. Covalent targeting is again appropriate when your target has got a reactive nucleophile, typically a cysteine or as with GPX4, a selenocysteine near the binding site. The covalent drugs form a permanent or slowly reversible bond which offers advantage in potency and duration. Targeted protein degradation. PROTACs, molecular glues. It is the approach for targets where inhibition is insufficient or where the scaffolding function of the protein matters independently of its catalytic activity. Degraders are particularly relevant for stress transcription factors like NRF2 and HSF1. Stress amplification deserves special mention because it is conceptually distinct from all the others. Instead of blocking an adaptive pathway, you increase the stress load on cells that are already near the threshold. That is the logic behind certain ROS amplifying agents, proteasome overloaders, and iron redox cycling compounds. The disease cell is already fragile, so you do not remove its crutch. Rather, you increase the weight it is trying to carry. Match the modality to the mechanism. This is the simple principle. Now coming to removing adaptive buffering. So it is the first of the two major therapeutic strategies in stress-targeted drug designing. The logic is very simple. Identify the pathway that is buffering the disease cell's chronic stress load, the pathway without which the cell stress state would become unbearable and again inhibit it. The clearest example in the literature is proteasome inhibition. Myeloma cells and certain lymphoma subtypes produce extraordinary quantities of misfolded immunoglobulins. They are under massive, permanent proteotoxic load. The survival depends on the proteasome continuously clearing this misfolded protein burden. Bortezomib removes that buffering capacity. Without the proteasome, misfolded proteins accumulate past the tolerable limit. The UPR activates its pro-apoptotic programs. The cell dies. Normal plasma cells, which do not produce this volume of misfolded protein, are thereby spared because they do not depend on maximal proteasome activity for survival. The same logic applies to HSP90 inhibition in tumors driven by client oncoproteins like HER2, EGFR mutants, ALK fusions that are conformationally unstable and dependent on HSP90 for proper folding. HSP90, if inhibited, and as a result, the oncoproteins misfold and thereby degraded. As a result, the tumor loses both its growth driver and its proteostasis support simultaneously. Removing the adaptive buffering is best suited for targets that are selectively required in the disease context, where genetic or pharmacological evidence confirms that the buffering pathway is essential to the disease cell but dispensable in the matched normal tissue. The second strategy, however, is in some ways much more elegant and in some ways more dangerous as well than removing just the buffering. It is the strategy of pushing the system past tolerance. So, instead of blocking the adaptive pathway, you have to increase the stress input. You add to the load that the cell is already struggling to carry. The disease cell, living near its adaptive threshold, cannot absorb the additional stress. It crosses the threshold. It collapses. Normal cells starting from a lower baseline, however, absorb the same perturbation and survive. This is the mechanistic rationale for pro-oxidant therapies in certain cancers. Tumor cells that have activated the NRF2 and upregulated glutathione synthesis to manage their chronic ROS burden are paradoxically more sensitive to agents that amplify ROS because they are already closer to the point where the antioxidant defenses fail. This is also the similar logic behind ferroptosis induction. A cell with high iron load, high lipid peroxidation activity, and active GPX4 defense is already living near the ferroptotic threshold. Inhibiting GPX4 or even depleting the sustained supply that sustains the glutathione synthesis tips it over. The cells with lower baseline iron and lipid peroxidation activity are further from the threshold and hence survive. The elegance of this strategy is that the disease state itself provides the selectivity. So, we don't need to target something that is unique to the tumor cell. You need to target something that the tumor cell specifically cannot tolerate losing because its baseline is already perilously high. The danger is very obvious. If the margin between the normal cell threshold and the disease cell threshold is too narrow, your therapeutic window is too small for clinical use. That is why biomarker selection is not optional, it is mandatory for threshold-based strategy. This slide consolidates the three major stress axes we have now examined from a therapeutic angle, and I want to use it to draw explicit connections back to my previous lecture. The UPR, coordinated by PERK, IRE1 alpha, and ATF6, it is the cell's response to ER proteotoxic load. In short, all three bunches work together. PERK reduces translation to limit the new protein input, IRE1 alpha expands the ER folding capacity, and ATF6 upregulates the chaperones. So this is the adaptive phase of the UPR. Chronically activated, the same system becomes maladaptive. CHOP, the transcription factor driven downstream of prolonged PERK activation, switches the program from survival to death. The UPR transition from pro-survival to pro-apoptotic represents a natural threshold built into biology. Therapeutic strategies that prevent the switch, keeping the UPR in chronic but non-lethal adaptation, may be neuroprotective. Strategies that force the switch, keeping chronically adapted cancer cells from ever returning to the baseline, may be anti-tumor. The NRF2 keep one axis is the master regulator of antioxidant defense. Cancers with KEAP1 mutation or NRF2 amplification have essentially hardwired their antioxidant programs on. They're protected from their oxidative damage, from chemotherapy, from radiation. So this is one of the most important mechanisms of therapy resistance as seen in many solid tumors. Targeting NRF2 directly becomes very difficult. Rather, targeting its upstream regulators, its synthetic lethal partners, or in fact using NRF2 activity as a patient stratification biomarker are the current approaches under investigation. Lastly, ferroptosis defense centered on GPX4 and the cysteine glutamate transporter SLC7A11. It is a threshold sensitive survival control. Cells with high iron, high fatty acid content, and high lipid peroxidation activity are already pre-sensitized to ferroptosis. GPX4 inhibition or SLC7A11 blockade in these kind of cells does not just reduce an adaptive response. In fact, it triggers membrane disintegration and irreversible death. This kind of threshold sensitivity is precisely the kind of vulnerability we want. I want to make a point that is often underappreciated in early target validation work and that becomes painfully apparent in late stage clinical failure. Cells do not organize their stress responses as independent modules. The UPR, NRF2 axis, ferroptosis defense, mTOR, autophagy, these are not separate programs running in parallel. They are the nodes in an interconnected network with extensive crosstalk, shared regulatory proteins, and overlapping transcriptional programs. So, perturbing one node has to change the state of many others. Inhibiting the proteasome activates the autophagy as a compensatory degradation route. Inhibiting GPX4 increases the ROS, which in turn activates the NRF2 that upregulates glutathione synthesis, which partly compensates for GPX4 loss. In the same manner, inhibiting mTOR activates UPR because mTOR restrains translation and its inhibition temporarily increases the translational stress. So, I have to emphasize that this network interconnectedness is the molecular basis of adaptive resistance. When you target a single node, the entire network has to reroute. The cell obviously finds another path to survival. This is not a failure of the biology, rather, it is a feature of the robust, evolved system. But what this means for drug discovery is that single agent programs targeting a single stress node will almost always face adaptive resistance. Effective intervention must reflect systems logic. You need to identify the network architecture, which nodes are highly connected, which compensatory routes exist, which combinations close off the escape routes before resistance emerges. And that is why the field is moving towards combination strategies, not as an afterthought, but as a first line design principle. Now, we should always remember that resistance is not a surprise, rather, it's an exception. But when you design a stress targeted program, your first question about resistance should be through which route the resistance is being developed. There are two architectures of adaptive resistance in stress networks. One is the interdisciplinary architecture, where multiple networks interact together to create a single resistance mechanism. Common resistance mechanism to single agent stress targeting. The second is the cell state switching. Under sustained therapeutic pressure, cells can undergo transcriptional reprogramming, that is, switching to an alternative cell state that is less dependent on the targeted pathway. Epithelial to mesenchymal transition in the carcinoma cells is partly a resistance mechanism that reduces the dependency on certain EF stress support pathways. So these kind of state switches are particularly difficult to anticipate and hence block. The rational response to both mechanisms is combination design. Like vertical combinations, they block the same pathway at the multiple nodes. For example, PERK inhibition combined with an upstream ISR kinase inhibitor, making compensation within the pathway harder. Again, horizontal combinations block the compensatory parallel route before it gets activated. For example, simultaneous proteasome inhibition and autophagy inhibition. The key is that the combination design must be guided by the network architecture, not by empirical observation that the two drugs together are better than one. You want mechanistically motivated combinations where the second agent closes a predicted escape route and not serendipitous synergy. Here we are approaching the end of this lecture and this series. I want to close with what I consider the most practically important point in translational drug discovery. You can have the most elegant biology, the most compelling genetic validation, the most potent molecule and still fail clinically if you cannot measure what you are doing in the patient. A target is not clinically real until it is measurable. So let me break that down. Target engagement means it demonstrates that your drug is actually binding its intended target in the patient's tissue, not in a cell line, not in a mouse, in the patient. So this requires pharmacodynamic biomarkers, that is the assays that read out the target occupancy or direct biochemical consequence of target engagement. If you cannot show this, you cannot distinguish between the drug didn't work or rather the drug never reached or engaged its target. Next comes the pathway modulation. It means demonstrating that engaging the target changes the downstream biology you intended to change. For example, if you inhibit PERK, can you measure the reduced EIF2 alpha phosphorylation? Or even if you inhibit GPX4, can you measure the increased lipid peroxidation biomarkers? Pathway biomodulation biomarkers connect target engagement to biological consequence. Patient selection is the hardest and most important piece. Stress-targeted therapies work best in patients whose disease cells are already near the adaptive threshold. To select those patients, you need to have predictive biomarkers, molecular features measured before treatment that tell you whether a given patient's tumor will respond or not. Stress transcriptional signatures, baseline NRF2 activity scores, ferroptosis sensitivity indices, these are the tools under development. Build the stop or go criteria into your program even before you begin. If at defined milestones you cannot demonstrate target engagement, pathway modulation, and the evidence of patient selection enrichment, the program has to stop. Not because the biology was wrong, but because the translational argument has already failed. This slide is the complete argument of this lecture series condensed into 5 short steps. Stress creates adaptation. We have already seen how chronic stress activates the conserved survival circuits across cancer, neurodegeneration, and infection. Adaptation creates dependency. Cells that continuously rely on an adaptive pathway become unable to function without it. The dependency creates vulnerability. A pathway that is selectively essential in disease cells but dispensable in normal cells can be a therapeutic target. Structure enables intervention. A biologically important vulnerability only becomes a drug program when a molecule can access and engage it, guided by structural biology, alpha-fold, and medicinal chemistry. Lastly, selectivity and biomarkers enable the translation. The drug reaches the clinic only when the therapeutic window can be predicted in advance through pharmacodynamic biomarkers, predictive signatures, and patient stratification. So this is the complete pipeline, and the logic that connects each step is not chemistry, rather it is the biology. So I want to leave you with 4 statements and not conclusions, rather commitments. These are the commitments that define a rigorous stress biology approach to therapeutic design. The same pathways that keep the disease cells alive can be used to eliminate them. This means the biology of survival is also the biology of death. It depends entirely on where you intervene and with what precision. The goal is a selective collapse of pre-existing fragility. Here, you are not creating damage in a normal cell. Rather, you are finishing a collapse that the disease state had already begun. Precision comes from context and not from pathway popularity. The best targets are not the most published or in fact the most discussed. Rather, they are the ones that are specifically essential in the specific disease state of the specific patient in front of you. Stress biology becomes medicine only when the vulnerability is translated, and translation requires structure, selectivity, biomarkers, and the willingness to build in criteria for stopping as rigorously as criteria for proceeding. So this has been my final lecture in the series. I hope what you take away is not a list of pathways or drug targets, but a way of thinking, a framework for converting biological insight into therapeutic action, a logic that starts with stress, moves to adaptation and dependency, and arrives, if the biology is right and the translation is done carefully, at a medicine that finally works. Thank you.
Thank you, Shreya, for that well articulated lecture. Our final lecture for today will be delivered by Jasmine Steffier. Welcome, Jasmine.
Good afternoon, everyone. I'm Jasmine Steffi, and today I'll be presenting on drug discovery fundamentals from biological targets to therapeutic designs. This presentation will walk you through the complete scientific pipeline of drug discovery, starting from identifying the disease-relevant biological targets, all the way from designing therapeutic agents and validating them for human use. And I'll also be highlighting on modern advancements, including artificial intelligence, precision medicine, and structure-based drug design, which are shaping the field right now. So, moving on, why drug discovery matters. Drug discovery is not a specific or just single step scientific process. It is a response to the growing global crisis. Different diseases like cancer, neurodegenerative diseases like Alzheimer's disease, alarming rate. Despite decades of research, many of these conditions still lack effective therapies. From a mechanistic standpoint, the challenge lies in biological complexity, and these diseases often involve multiple dysregulated pathways, not just one, rather than a single retreat. Another major issue is safety and specificity. The traditional drugs often interact with multiple unintended targets, leading to adverse effects. Therefore, modern drugs that are discovered today emphasis on precision targeting, minimizing off-target toxicity, and incorporation of several carriers for targeted delivery. Additionally, advances in genomics, structural biology, and disease biology by incorporation of machine learning mechanisms have expanded our understanding about that disease and its mechanism, have opened new opportunities for us to identify the therapeutic intervention that is required at this moment. And what is drug discovery? Drug discovery is a multidisciplinary and an iterative process aimed at identifying compounds that can modulate biological systems to produce different therapeutic effects. It integrates multiple domains of life sciences, starting from molecular biology and cell biology for understanding the disease mechanisms, medicinal chemistry for designing a particular compound, pharmacology, toxicology for evaluating biological effects, pharmacodynamics is also involved in this, and computational sciences for predictive modeling. From a timeline perspective, this process typically takes around 10 to 15 years, which costs some billions and billions of dollars. And most importantly, only about 1 in 10,000 compounds successfully reaches the market, crossing all the barriers, starting from the in vitro testing to the in vivo testing and the animal testing, and followed with the clinical trials. It's really 1 in 10,000 compounds that successfully reaches the market. And the drug discovery pipeline. Drug discovery follows a stepwise funnel like pipeline. There is filtering out of different candidates at different levels in this pipeline. Generally, we begin with target identification, target validation, followed with lead discovery, lead optimization, and then followed with the preclinical studies. At each of the stages, different compounds are identified, are filtered based on its efficacy, selectivity, pharmacokinetics, and safety. There are innumerable number of variables that's involved in each of these steps. This funnel ensures only the most promising candidates proceeds to expensive clinical trials. So this is followed with identifying of the drug targets. A drug target is typically a biomolecule which can produce modulations, like which modulates a therapeutic effect. The most common targets often include proteins and enzymes such as kinases, which are heavily involved in cancer signaling. These kinases are a key enzymes involved in phosphorylation, which turns on accounts of several pathways in cancer, and receptors, especially GPCR and ion channels that regulate cell communication. Genes and nucleic acids also plays an important role by increasingly using antisense technology and gene therapy. The choice of target is critical, but it also determines the mechanism of action of the drug. And target identification. For the target identification, there are several methods used by several scientists all across the globe. different approaches like genomics, proteomics, bioinformatics, and disease biology. Genomics is generally identifying differentially expressed genes. It involves gene expression profiling, studies, and transcriptomics. These reveals which genes are upregulated, downregulated, or silenced in disease states, flagging the potential molecular drivers in this part. And proteomics gives us the mapping, like overall mapping of protein interaction and its modification. It is generally done with the help of mass spec based protein profiling, and it changes in protein abundance, post translational modifications, and the interaction between the network of proteins in diseased as well as healthy tissues. And further moving on, now we'll go with bioinformatics. Bioinformatics is an integrated approach integrating multi-omics datasets, network analysis, pathway enrichment tools, and machine learning algorithm integrate multi-omics data to prioritize high confidence target candidates. Disease pathway analysis. This involves identifying key signaling nodes. For example, dysregulations of PI3K or AKT or mTOR pathway is commonly implicated in cancer, making it a valuable therapeutic target validation. Target validation, it's once a target is identified, it must be validated to confirm its functional relevance. This involves several studies, starting from gene knockout studies, RNA interference techniques, CRISPR-Cas9 editing, animal model, and etc. Validation ensures the modulating target actually produces a therapeutic benefits rather than merely correlated with the disease. Initially starting with the gene knockout studies. In gene knockout studies, the silencing or deleting the target gene in cell line reveals whether the loss of a function of the phenotypes disease or produces a beneficial effect. Then RNAi technology. This approach transiently suppresses target expressions, rapidly assesses the functional, enabling rapid functional assessment without permanent genomic alteration. CRISPR based models, as we all know, it's a very precise genomic tool, creates isogenic cell lines and animal models with defined targeted mutations for mechanistic studies, followed with animal models, in vivo validation, and transgenic or pharmacologically induced disease model. Test whether the target modulation translates to the whole organism benefit or it is localized. Lead compound. A lead compound is a molecule that shows measurable biological activity against the target. And these lead compounds can be a small molecule, small peptide, or a biosimilar. This biological entity that demonstrates measurable activity against the validated targets. These targets can be any receptor or any ion gated channels or like that in cancer at an acceptable concentration. Leads are not finished drugs, obviously. They are promising starting points that require extensive optimization before clinical evaluation. Key characteristics of this lead compound involves potency. It's often measured as IC50, and selectivity of the target, drug-like properties, which is often guided by Lipinski rule of five. But however, these leads are not final drugs, as I said earlier. These require extensive optimization. While selecting the lead compound, different biological activities like as is it showing reproducibility, concentration dependent modulation of the target, and the selectivity of the profile shows preferential activity against the intended target over closely related off target, and drug like property, molecular weight, lipophilicity, hydrogen bonding as a predictor for bioavailability. Lead discovery methods. So high throughput screening is one of the method, natural product screening and virtual screening. These are the three commonly used late discovery methods. Um, high throughput screening, which tests thousands of compounds rapidly, natural product screening, which has like historically yielded drugs like antibiotics, phytochemicals from different plant, and virtual screening, which uses computational models to predict binding. Each method has its own advantages as well as disadvantages depending on the target and availability of the resources. Once the lead is identified, it is followed with a very important step called lead optimization. Lead optimization represents a critical stage in drug discovery where a promising lead compound undergoes systematic refinement to enhance their therapeutic potentials. Lead optimization is an iterative process. It goes on and on and on. A medicinal chemist modifies the molecular structure to increase binding affinity, reduces toxicity, and improve metabolic stability. The stage involves balancing multiple parameters from pH, temperature, from different bondings, from starting from hydrogen bonding to covalent bond, CC bond, CO bond, and often requires several design test cycles. So this is mainly aimed to increase efficacy, improve stability, and reduce toxicity, enhanced binding affinity to the target proteins, and improve biological activity through structural modifications that modify molecular interactions. Reduced toxicity often to eliminate or minimize the off-target effects and adverse reactions by modifying chemical moieties that might cause unwanted biological interactions. And the last one is to improve stability. They enhance the metabolic stability and shelf life through structural changes that resist degradation pathways and maintain the compound's integrity after consumptions. Structure based drug design. Structure based drug design, or SBDD, uses the 3D structure of the target proteins to design drug rationally. There are different techniques used here like X-ray crystallography, cryo-electron microscopy, molecular docking, and simulations. A major breakthrough in this field is AlphaFold 2, which predicts protein structure with a very high accuracy. SBDD significantly improves efficiency and selectivity in drug development. This SBDT has its own set of advantages. SBDT significantly improves hit rates compared to blind screening, shortens the optimization cycle, enhances selectivity by designing around like very unique features of the target binding site. It is now a cornerstone of modern drug discovery workflow across academia and pharma industries. This AlphaFold 2 has made SBDD accessible to even for targets lacking experimental structures, where the database doesn't exist, opening a new frontier in rational design. ADME properties. The ADME properties actually defines how a drug behaves inside the body. A, that stands for absorption, decides how the drug enters the bloodstream through various routes, through starting from oral, intravenous, transdermal administrations. Then the D, that stands for distribution, the transport of drug throughout the body and its partitioning into the different tissues and organs. Metabolism, enzymatic transformation of drug into metabolites, primarily occurring in liver through phase 1 and phase 2 interaction. And the E, that stands for excretion, removal of the drug and its metabolite from the body via kidney, liver, lungs, or other elimination pathway. ADME profiling is very important and it's a cornerstone step in the modern drug development process, encompassing four interconnected processes that determines the compound's fate within the body. Understanding of these properties enables the researchers to identify and predict themselves the effectiveness and the optimizing those strategies. Toxicity screening. Toxicity screening is conducted early to eliminate harmful compounds. This includes cell-based assay, cytotoxicity assay, functional assays. Early identification of toxicity helps reduce failure rates in later stages. Toxicity screening actually stands as the first line of defense in identifying potentially harmful compounds before we move on with costly and expensive studies onto animal. This critically evaluates the quality and employs a battery into the in vitro assays designed to detect cytotoxicity, genotoxicity, and organ specific toxicity. Basically, in vitro cell based assay, we utilize human cell lines to evaluate compounds' effect on cellular viability, proliferation, and function without any animal testing. And cytotoxicity testing measures cell death and damage through multiple endpoints, including membrane integrity, metabolic activity, and apoptosis marker. Early safety evaluation, it identifies red flags in a compound series, allowing medicinal chemists to redirect the synthesis. And elimination of harmful compound helps saving resources and preventing advancement of unsafe compounds to critical clinical development. Preclinical studies bringing laboratory and clinic together. Clinical trials are conducted in three phases. Phase 1, generally to identify the safety and dose in healthy volunteers. Phase 2 is to identify the efficacy in patients, and phase 3 is large scale validation. Each phase is progressively more complex and expensive. And in first stage, preclinical study, safety assessment, which evaluates acute chronic toxicity, identifies maximum tolerated doses, and characterizes adverse effect across different organ systems. Followed with pharmacokinetics, characterizes ADME properties in vivo, determining half-life, bioavailability, clearance rate, and distribution pattern among the tissue. Toxicology studies conducts comprehensive safety pharmacology assessing cardiovascular, respiratory, and central nervous system effects. Regulatory approval. So, regulatory approval is a gatekeeper of patient safety. Regulatory approval represents the culmination of years of research and millions of dollars in investment. Government agencies that includes FDA from United States, EMA from Europe, PMDA from Japan, and CDCSO from India evaluates comprehensive dossier documents in every aspect of drug development. Approval requires demonstrating that benefits significantly outweighs the risk for the intended indication. Regulatory authorities scrutinizes the manufacturing process, quality control system, quality assurance system, and post-market surveillance plans before granting any marketing or selling authorization. This has three different components: safety, efficacy, and quality. These three has to be met before the compound reaches the market. Comprehensive adverse event profile demonstration, acceptable risk benefit ratio across diverse patient population, efficacy for statistically significant therapeutic benefit compared to placebo or standard of care in controlled clinical settings, and quality, consistent manufacturing processes, practices ensuring drug purity, potency, stability throughout shelf life. Modern drug discovery. Modern drug discovery is constantly evolving and it is rapid. From precision medicine to biologics to nanotechnology and artificial intelligence. Tailored therapeutic strategies to individual genetic profile is precision medicine. It enables targeted intervention for patients with subpopulations with specific molecular characteristics. When we see about biologics, it utilizes the monoclonal antibodies and recombinant proteins as therapeutics, offering high specificity and novel mechanism of action for previously undruggable targets. Next comes the newer part of the drug discovery part, nanotechnology. Engineers engineer nanoparticles for targeted drug delivery, enhancing therapeutic index while minimizing the systemic toxicity through site specific payload. Followed with artificial intelligence, which employs machine learning algorithms to predict the compound properties, optimizes the lead structures, and identifies novel target through different pattern recognition in massive datasets. Different challenges that's faced during drug discovery. Despite several advancements and several milestones that are reached in today's world, major challenges still remains. Drug discovery remains as one of the most complex and resource intensive, investment intensive in modern science. Understanding these challenges is essential for settling realistic expectations and developing robust development strategies. First, it is a time intensive process and it requires high development costs, elevated failure rates, and regulatory complexity. Time-intensive process from target identification to market approval typically requires 10 to 15 years, demanding sustained investments and long-term strategic planning across multiple development phases. High development cost. Average cost exceeding 2.6 billion per approved drug, including failed candidates, driven by extensive testing, regulatory requirements, and clinical trial complexity. Elevated failure rates. Approximately 90% of the drug fails to reach approval, with attrition occurring at each development stage due to efficacy, safety, or commercial viability concerns. There is also regulatory complexity. Evolving requirement across different continents, different countries demands specialized expertise and extensive documentation, with regulatory strategies requiring continuous adaptation. Drug discovery is a multi-step process. Here to summarize the entire presentation, drug discovery is a multi-step, highly regulated process that integrates biology, chemistry, computational science, and bioinformatics. Each stage, from target identification to clinical trial, acts as a filter to ensure that only safe and effective drug reaches the patients. And it is very important that in pharma industry that is involved in the identification of a drug or marketing of a drug follows through it, from target identification, lead generation, lead optimization, and preclinical development and clinical trial. This process demands collaboration among high potential investors to honest and hardworking scientists. The future of drug discovery, as we discussed earlier, personalized medicine, AI driven drug discovery, and advanced therapeutic systems are going to be the future. These are already into existence and these are going to take over the future of drug discovery. It is being reshaped by technological breakthroughs, I would say, and it's seeing a shift in therapeutic development. Emerging approaches promises to accelerate timelines, reduce cost, and deliver more effective and targeted intervention. For example, with the COVID vaccines, personalized medicine, integration of genomics, proteomics, and clinical data enabling patient stratified therapies with optimized benefit risk. AI driven drug discovery, machine learning algorithms predicting compounds property, generating novel chemical matter, and optimizing developmental strategies. Advanced therapeutic system. Next generation modalities, including gene therapy, cell therapy, RNA based therapeutics, expand the drug will target universe. The convergence of artificial intelligence, precision medicines, and advanced therapeutic modalities heralds a new era in drug discovery, transforming how we identify, develop, and deliver life saving medicine. On closing note, I would like to say and drug discovery represents one of the most complex and an important, impactful area of modern science, with the potential to transform global healthcare. With this, it requires high attention. Thank you for your attention. I'm happy to take.
Thank you, Jasmine, for that comprehensive lecture. Now, as we conclude today's scientific sessions, we now shift from scientific concepts toward academic and research journeys. One of the important aspects of building a career in science is not only understanding research topics, but also about learning how to navigate opportunities in higher education, PhD applications, mentorship, and global research environments. To share these insights on these areas, we are joined by two distinguished researchers and mentors, Dr. Brooks Robinson and Dr. Alfredo Gezzi. Before we move into these mentor talks, I would like to thank all our participants and audience for making today's session enriching. We hope these sessions continue to encourage curiosity, deeper scientific thinking, and interdisciplinary learning among everyone watching. We also look forward to welcoming you to day 4 on 13th May, where the sessions will focus on translational drug discovery along with bioinformatics and computational biology in modern biomedical research. With that, we now move into the mentor talks. Thank you once again for joining us today.
What if I told you that getting into your dream PhD program wouldn't come down to your grades or even your research experience, but to how well you play the game? Hi, my name is Brooks Robinson, and I've been on both sides of the table. I've applied to PhD programs myself, but I've also reviewed PhD applications, mentored students, and watched brilliant minds get overlooked because they didn't know the rules of the game. Let's face it, most students don't fail because they're not smart. They fail because no one taught them how the system really works. I'm part of a team of professors, scientists, and experts led by Dr. Sukan Karana. We've helped students from all over the world get into top PhD programs in Germany, France, the Netherlands, and beyond. We know what works: how to write a letter that lands an interview, how to email professors and get responses, and how to write a CV that helps you stand out from the hundreds of other applicants. This is not guesswork. This is strategy from the other side of the table. Instead of stumbling through the process, learning from your mistakes, learn from ours. We've reviewed hundreds of PhD applications and helped shape even more. Why go it alone when you can be guided by the people who make the decisions? If you're serious about a PhD in Europe, stop guessing. Email us at contact@ioncureRx.com with your CV and goals, and let's build you an application that gets results. Because success isn't just about hard work, it's about smart work with the right team. Join the students who've made it because they have the right mentors. You could be next.
Hello, I'm Alfredo Gezzi, professor and director of a graduate program, and someone who has spent years reviewing PhD applications from around the world. Let me tell you something honestly. Far too many brilliant students don't make it through the door, not because they lack potential, but because of avoidable applications. Here are just a few common issues I see again and again. A generic motivation letter that could apply to any lab, anywhere. A CV that reads like a shopping list, not a story of academic growth. Students who fail to connect their past work to the lab they're applying to, and surprisingly often, poor communication with the potential supervisors. These things may seem small, but trust me, they are deal breakers. But here's the good news. You don't have to figure this out alone. I work closely with Dr. Sukan Karana and an international team of professors and scientists. We have sat on admissions panels, we've guided hundreds of students, and now we're here to guide you. We understand what admissions committees are really looking for, and we are ready to help you present your story in the best possible light, tailored, strategic, and authentic. So, if you're serious about pursuing a PhD, especially in Europe or North America, don't leave it to trial and error. Reach out to us at contact@ioncurex.com. Let's work together to make your application not just good, but unforgettable.